everybody, and welcome to our workshop on AI for health, applying AI to model and understand multimorbidity. My name is Wojciech Samek. I'm AI researcher and professor for machine learning at Technical University of Berlin. And I'm very happy to co-organize this workshop with Birgit Sawicki. And I mainly work on explainable aspects of machine learning methods, which allow us to understand what deep neural networks and other machine learning models have learned. And I'm convinced that this combination of understanding, explaining, interpreting our models, and the amount of data which we have, especially in the health domain, can help us in the future to better understand diseases, better make predictions, better manage and learn something about complex diseases such as multimorbidity. So I very much look forward to discussions with you about this topic of AI for health. And I would like to give the word to Birgit.
Yeah, so welcome also from my side. I'm happy to see you all here, and also very happy to introduce our topic. I'm heading together with Wolfgang Kübler, who will be the first speaker of this workshop, an initiative we are applying for the most prestigious grant in Germany, funded by the DFG, or German Research Foundation, so-called excellence cluster. And we came up with an idea, a topic which is really demanding, and that's multimorbidity. Multimorbidity, as Wolfgang will also explain, is by definition the occurrence of several diseases, life-threatening diseases or major events in a patient. And at the moment, the patients are only managed, but there's no mechanistic understanding on how this develops and how we can prevent development of multimorbidity. And because the prevalence is increasing, there's a huge medical demand. And the data we are dealing with that topic here is so huge that we need to apply AI approaches to really develop in a fast manner solutions to help the patients. And that applies different aspects of our population. And we will hear that by the different speakers we have gathered together from our consortium, which they will address, like also gender aspects or modeling in certain in vitro models. I didn't introduce myself. This is what I usually forget. So I'm Birgit Sawicki. I'm the chair of Professorship for Translational Immunology at the Berlin Institute of Health, which is the part integrated in the Charité, what they always say, Europe's largest university hospital, only because of the numbers of beds, I would say, but oversees a huge patient population, and that's why we also think this is well suited there at the located at the Charité, because we are overseeing so many patients and also patients with multimorbidity. And immunology plays, and the immune response plays a very important role in driving injuries to several organ systems. And that's why I also see a role for myself in that topic. And without further ado, because what we would like to do is to engage into discussions on how one could apply AI approaches to accelerate our understanding of multimorbidity, I would introduce Wolfgang Kübler, who will tell you a little bit about our concept and why we think we have a cool hypothesis which can help to understand multimorbidity. He is the head of the physiology department at the Charité, and therefore also physiology is always at the heart of the function of all organ systems, well placed as the co-speaker of our initiative. So Wolfgang, please
Thank you. Thank you very much for the introduction, Birgit, and Wojciech, and thank you very much for the invitation and for having me here. So my task today is to set the stage, to define the problem, and then I will leave it to the experts and to you to see how, with this big data that really emerges with this problem and with these analysis that we're facing, may be analyzed by the application of AI. So we're talking about multimorbidity. And as Birgit already mentioned, multimorbidity is really defined as the coexistence of two or more acute or chronic disease conditions in one person or in one patient. And multimorbidity is really, as you will see in the next slides, in sort of the next pandemic. It's a really strongly emerging topic with high incidences and high prevalences. And that goes around the entire globe. It affects low, middle, and high income countries in a similar way. And it imposes major demands and costs on the healthcare systems. And largely because we currently have no good concepts how multimorbidity develops. And because we don't have good concepts, we have no known risk factors, we have no way to predict it, we have no prevention concepts, and most of all, we don't have therapies really to treat it. What you see on the right is just illustrating this emerging pandemic, as I call it. This is multimorbidity assessed in England in the early 2000s and in 2019 on the right side. In 2004, approximately 15% of the English population had three or more chronic diseases at the same time. 30% had two chronic diseases at the same time, defined as multimorbidity. 15 years later only, these 30% of two diseases had increased to 50%, and the 15% of population which had three or more diseases had increased to 30%. So that's a dramatic development, only almost a doubling of numbers within only 15 years, and you can see where this is going. Why is it increasing so fast? Well, one factor, probably not the only one, is that multimorbidity is very strongly associated with age. And of course, we live in many countries in aging populations. We have demographic shift. And you see here how, with increasing age on the x-axis, the number of chronic diseases that coexist in one person, in one patient, increases dramatically. I said before, it's a global problem and it affects all countries, but unfortunately, it's also one of these problems that particularly affect those who are less fortunate in terms of their socioeconomic status. So people with a low socioeconomic status, which are here given by high numbers, 9, 8, 10 on the right panel, they have an increased prevalence again of multimorbidity as compared to people with a socioeconomic status. So how does multimorbidity really develop? And the prevalent view right now is very simple: coincidence. So you happen to have a disease emerging in one organ, like shown here for the kidney, which slowly, as you can see here, turns from healthy gray to unhealthy red, from the left to right, and at the same time, for purely random reasons, maybe the heart also gets a disorder. And that's how different disorders in different organs accumulate over time in the same patient. But that's probably too simple. If you look at big population-based databases, and this is data from a big Tokyo study on all patients who received medical care in Tokyo, aged over 75 years, over, I think, several years time span, was more than 1 million patients included. They listed all the different diseases that occurred according to age, and you see here the number or the percentage of patients with these diseases under all for men and for women. And if you look at these individual diseases and then for the combinations of these diseases in the patients, you see that there is not just random clustering. For example, if you look at completely seemingly independent diseases in men, like hypertension, peptic ulcer disease, and urologic disease, complete different organs, seemingly fully unrelated. Based on the incidences of each of these diseases, you would expect a random clustering of all three diseases in one patient in a bit less than 6% of patients. However, it's more than that, it's 8%. Yeah? Same for women. If you take three exemplary diseases, again, hypertension, peptic ulcer, and here insomnia, which is more frequent in older women in Japan than in men, again, you have more than random distribution of an accumulation of these different diseases. So there's something happening here which is not just coincidence. And we can see that across many population-based studies, and that has led to the concept that these diseases cluster in certain groups. This is data from Scotland, which looked at all patients hospitalized in a hospital for chronic disease treatment, and they tried then to see which of these diseases would appear more than randomly together in groups and called that clusters. And you can see that some of these clusters intuitively make sense. For example, hypertension, yes, with atrial fibrillation and flutter. So diseases of the heart and the cardiovascular system belong together. I think it's also sort of intuitive that alcohol misuse and depression may go along with each other. But then you have respiratory diseases such as asthma and high blood pressure, hypertension going along with each other, which doesn't intuitively make sense. That's different organ systems. So how does that link together? Yeah. And these clusters exist across multiple cohorts and again involve very different organ systems. This is pretty small, unfortunately, I apologize, but this is again from a population-based study in London, more than 1 million people seen by general practitioners in the community over different years and time spans, as you can see in these different panels. And you can see that there are different clusters of diseases highlighted here in different colors, but also very different diseases that seem to cluster here. If you look at the green cluster here, chronic obstructive pulmonary disease, COPD, so respiratory disease, together with diabetes, which is here DM, and osteoarthritis, so joint disease. And at the same time, I think you also have in the same cluster here, sorry, I can't hardly see it from this side, hypertension and pancreatic and Parkinson's disease, so neurological disorder. So a very diverse set, which seem, however, to be somewhat related. Yeah. And I think that's something we have to keep in mind. And with that in mind, we thought we need to change the prevalent view of this random occurrence of diseases in multimorbidity and replace it with a more adequate concept accounting for these interactions which seem to exist between different organs. And the way that this probably happens, and that's the emerging view on multimorbidity, and I'm going to illustrate why we think and what the evidence is for that, is that organs, of course, don't act in silos and in isolation. Yeah, in principle, we all know that. Yeah, but they talk to each other constantly. They talk to each other in a healthy body system. Yeah, and maintain each other's healthy status, which we call homeostasis. But also, when one organ becomes injured and damaged, then this inter-organ crosstalk, this homeostasis, becomes disturbed and dysregulated. And that results then in negative effects on the other organ and can kick off an avalanche that then affects many different organ systems and promotes the development of what we call multimorbidity. I said I'm going to show that this is actually indeed happening by some epidemiological evidence. This is evidence from studies looking at the coincidence of cardiovascular disease and pneumonia, seemingly completely unrelated diseases, one affecting the lung, one affecting the cardiovascular system. Yeah, and many of you probably had pneumonia once, or you know somebody who had pneumonia. Bad news, it's bad for your cardiovascular system. And it's not only bad for your cardiovascular system when you have pneumonia, because then you take antibiotics and usually you're fine. It actually affects your cardiovascular system for the next 10 years to come. Yeah. Having had pneumonia once in your lifetime is as bad a risk factor for your cardiovascular system as smoking a pack of cigarettes a day, having high cholesterol in your blood, or having high blood pressure, unfortunately. If you have had one episode of pneumonia and one additional cardiovascular risk factor, your risk for having a cardiovascular event, so a myocardial infarct or a stroke over the next 10 years, is almost 60%. If you happen to have one episode of pneumonia 10 years ago, and you happen to have two cardiovascular risk factors, unfortunately, your risk of having a stroke or myocardial infarct is almost 90%. If you didn't have pneumonia, it's only 25 or 30%. Yeah? So there is some big interaction going on there between this lung infection and the cardiovascular system that we don't understand at all. Yeah? That's not only true for the cardiovascular system and the lung, that's true for many other systems. I'm mostly a lung and cardiovascular physiologist, so that's where I draw my examples from, but you can make the same case, for example, for pneumonia and renal disease. So chronic kidney disease is much more frequent in patients who had pneumonia. This is a population-wide study from Taiwan, which shows a higher incidence of chronic kidney disease in patients who had pneumonia, again, over 12 years. And also other studies, this is a Swedish national cohort study, show that the incidence of chronic kidney disease is significantly increased in patients who had so-called community-acquired pneumonia, so regular pneumonia, which is called CAP here. And this is completely linked to pneumonia as the primary diagnosis. It's independent whether the people had multiple episodes of pneumonia or not. It's also independent whether they already had kidney disease when they had pneumonia. No, it really develops as the pneumonia goes on. And it's also independent whether the pneumonia resulted in a sepsis, so in general bacterial spread through the organs, which would of course explain why it goes to the kidney. No, that's not the cause, even if the pneumonia only remains in the lung. People get chronic kidney disease. Nobody knows why. So there seems to be something at play here, that disease of the first organ, here highlighted by the lung, somehow sends a signal, yeah, to the other organs, which then causes health or disease in the other organs, here shown by the heart. And that's sort of a classic communication pathway signal with a sender, a signal, and a receiver. Of course, in real life, in the healthy organism or in the diseased organism, this is in fact much more complex. Yeah, we have a sender and a signal transduction from the sender to the receiver, but this sender can send signals in many ways. Yeah, you can have, as in the upper example here, just written under channel, just a signal released with these green dots, which goes to the receiver. The signal can, of course, also be disturbed by other signals, so-called noise, or it can be altered on the way from the sending organ to the receiving organ by what we call relays. Yeah? And these changes in signal can be amplifications, as you see here in the relay signal that suddenly increases in the number of green dots, or it can be changed entirely. I will show you examples how that would work. And the whole process is, of course, constantly undergoing modification by modifiers, age, sex, environmental cues, etc. And also, as shown on the bottom, usually underlies feedback mechanisms. So the receiving organ will again send signals back to the original diseased organ. But it makes the whole thing already very complicated. You can see where we're going in terms of big data and how to analyze this. Yeah, it becomes even more complicated because we have to look at very, very different modalities and dimensions of these signals. Yeah, because these signals can be proteins like classical hormones, they can be peptides or small amino acid chains, they can be lipids, they can be just metabolites. So one organ is eating up more of glucose or glutamate, as shown here as an example, and therefore the other organ gets less of it. Can be little small parts of cells which are released, so-called extracellular vesicles, which release genomic information, but also proteins and transport them to other cells. Can also be toxins, which can come from bacteria, but also can come from our own cells. Think of Alzheimer's disease, which basically are toxins released by our own body, which travel through the system. Of course, it can be neuronal signals which connect organs together. It can be mechanical signals, yeah, like for example, blood pressure or changes in flow, etc. So very different modalities which can come to play here. And as I mentioned before, these signals also become changed on the way from the sender to the receiver by so-called relays. What do we mean with relays? Well, a classic model for such a relay would be the immune system, yeah? And let's go back to our example of pneumonia causing chronic kidney disease. Yeah? If the bacterium goes into the lung, and he exemplified that here with Streptococcus pneumonia as the most common cause of pneumonia, then we elicit inflammation there, for example, by releasing toxins such as pneumolysin, which is a toxin released by these bacteria. This is sensed by the immune cells in the lung, so-called macrophages, which then react and respond and try to fight this infection by releasing inflammatory mediators, cytokines, such as this mediator TNF-alpha here. Yeah? This is released against the bacteria, but also is released into the bloodstream. Yeah? Now, in the bloodstream, it travels, of course, through the body, goes to the kidney. As you see in the kidney, there are other immune cells, such as this neutrophil, which you see there, which is another very active immune cell, which can then get activated through this TNF-alpha and produces very aggressive substances in order to fight against an emerging infection, even though the bacteria are not even in the kidney themselves. But that damages not only bacteria, it damages the kidney, and therefore you have kidney disease. So you see how the initial signal, namely the pneumolysin from the Streptococcus, becomes changed into other signals by these relays, which are immune cells. Other relays in our system are barrier cells, yeah, which basically prevent signals to disseminate across a barrier, but can also generate new signals. And classic example is the barrier in the lung, where, for example, a mechanical signal such as increased pressure in the blood system can then be sensed by the cells outlining these blood vessels, and they will respond. That's data that we have published 2 years ago, where we show that these cells respond by an increased formation of another mediator, so-called nitric oxide, which then signals to the adjacent cells outlining the air spaces, which then respond again to this signal with another mediator, so-called surfactant. So you see different cells can change the entity of these mediators acting as relay signals. So those are relays. What about modifiers? And I mentioned in the beginning from the epidemiological data, probably the most important modifier that we have is age in addition to sex. And I know that George Stadler is talking about sex and gender later on, so I leave that totally to her as the expert. But I'm going to talk a little bit about age as a modifier. And there's actually very exciting emerging data on that. This is a paper from last year published in Nature, where the investigators looked at the proteome, the plasma proteome, in a large number of different subjects. And from the signatures of organ-specific proteins in the blood, which they identified and which they validated and trained based on a machine learning approach in a large cohort of healthy patients with different age, to establish normal age profiles of these organ-specific proteins, then used and then validated in various cohorts shown here below, the Covens and the Longevity and SAMS cohort. They basically use these normal age protein signatures in the plasma then to calculate so-called age gaps for patients in certain organs. Yeah? So if your protein signature for liver proteins is older than your biological age, you have an age gap, yeah, for your liver. Yeah? And this is shown here on the right upper panel, this difference between this age gap and the chronological age. And that correlates actually nicely with really clinically evident disease in the respective organs. Yeah, so it's a predictor of this organ function in a way. Yeah? And what is interesting is that about 20% of patients in a large population study actually had increased biological age of individual organs as compared to their chronological age they should have. And this was highly predictive of morbidity and mortality that would occur in these patients. More importantly, if you take these individual organ ages together, you would think that multi-agers should be extremely rare because 2% of patients had advanced kidney age, 2% had advanced heart age. If you take 2% of 2%, you end somewhere at 0.04%. But the incidence of these multi-organ agers is actually almost 2% as well. Yeah? So again, if you have probably advanced aging in one organ, you are very likely of having advanced aging in other organs too, with respective consequences, of course, for this organ function. And that's supported by another study also published in Nature last year, which looked exactly at this interaction in aging in different organs. They didn't use protein markers in the plasma. They actually looked at functional data and imaging data from the brain and from many other organs in the entire body. And again, trained their system based on these data to recognize normal biological age and then identify age gaps. Yeah? And when they did so, they could actually calculate that if you have an age gap in one organ, how it would affect over time the rate of change in age gap in the other organs. Yeah? So if you have one organ which is advanced aged, how it will over time promote aging of the other organs. Yeah? And you see there's a massive influence. Yeah? If you have advanced aging in the lung, it will rapidly affect with 30% impact on the heart, on the muscle, on the renal system. The cardiac system, of course, is very much in the center of the whole thing. Very surprising for me, the relevance of the skeletal muscle as a central player also in this whole connectivity here. And you see how this all is connected through communications that we don't understand. We don't know what the signals are, but that are strongly modified by aging. This is just from last week, and I thought it just fits so perfectly to the topic that I had to take it in, a sort of science article in Science, just exactly focusing on that and saying how 40 messages between organs could make us old. Yeah? That's exactly what seems to be happening. It's not unique to aging, as we say, it's really what we think drives multimorbidity, but it's closely related to aging as a modifier. So how do we address this now? Yeah? Well, we have to find these signals. We have to identify why certain organs release these signals, why other organs may be particularly susceptible to these signals. And we can do that in unbiased approaches. One example for that is work here from Andreas Herrlich from Washington University, who looked in a mouse model about on the development of lung injury after an acute kidney injury. So they induced kidney injury in these mice and then sometime afterwards looked at their lungs, and they see that at different time points, day 1, 3, 5, you are used to looking at lungs, then you see that this lung structure starts to look not normal anymore. Yeah? Sham is sort of the healthy one, and that's what you want. And in the other ones, you see there's more tissue and more red stuff, and that's not good. Yeah? And what they then did is that they basically did a transcriptomic analysis, so gene signatures in the kidney to see what's happening here, and at the same time, similar gene signatures in the lungs in all different cells, so-called single-cell RNA sequencing, and try to correlate which signals are now upregulated in the kidney and which receptors for such signals are upregulated at the same time in the lung. And that can be analyzed bioinformatically by various softwares. One of them is this cell phone analysis. And by doing so, they came up with one signal that they identified, which they said is high in the kidney, and the receptors are high in the lung, and that could be a potential candidate. That's a molecule called osteopontin. And then they followed up and did targeted approaches where they knocked out osteopontin in these mice or neutralized it, antibodies, and in that they could prevent the emergence of lung injury. So that's one way to go in that approach. We did similar approaches in close collaboration with Markus Alser, who is an expert in proteomic analysis at the Charité in Berlin during the COVID pandemic, where we thought, how come that in COVID, as we all unfortunately had to learn over the last 5 years, you get multiple organ involvement from a disease, which is actually a virus affecting the lung. Yeah? In most of the cases, with few rare exceptions, the virus doesn't go through your whole body. Nevertheless, people, as we all know, get neurological symptoms, kidney disease, heart disease, whatever, following COVID disease. How does that happen? Well, it must be signals that are released there and disseminated, so you must find them in the blood. And so we took the blood from patients with various disease severities of COVID, and Markus did comprehensive proteomic analyses, and this is just highlighting a lot of different proteins that are massively upregulated as a function of COVID severity, which you see here on a scale from 3 to 7. And on the very left of each panel is a serum control, and on the very right side of each panel is the generation Scotland, which is a healthy population control. And when we then take this plasma, and now, Wojciech, you would have to press on, sorry, yeah, on, if you go on the image, or yeah. Yes, thank you. If you use this plasma and put it on endothelial cells, which outline our normal blood vessels, yeah, you see on the left side what happens to these cells if you put plasma from a healthy patient on it. The cells stick nicely together, they move a little bit around, yeah. But you can imagine if this is outlining my blood vessel is tight, yeah, it's sealed, yeah, there's nothing getting out between these different cells. But if you look to the right side, that's these cells now stimulated with plasma from a patient with COVID. And you see how they pull apart and how they form gaps between these cells. And that's where fluid now and everything that was in the blood gets out into the tissue. And these people get massive disease. You can imagine if this fluid goes into your lung, you can't breathe anymore. And that was, of course, the most prominent problem in these patients, but the same happening in other organs as well. And that's not related anymore to the virus. That's because of all these inflammatory proteins. And now you can go and identify which one is it, are related here. Yeah? And this just shows on the right bottom that this relates directly to the disease severity of this COVID disease, how this disruption works. How do we know where these signals come from? Well, in patients, it's very hard to tell. Some proteins, as I mentioned before, are organ specific. In other cases, we have to resort to non-clinical models. And over the few past years, there have been developments of techniques to actually label mark signals where they come from. This is beautiful work from Ilya Ruzhin from Boston at Harvard, who basically developed cells and models and mice also, which express a biotin ligase, which they originally also from bacteria, which they engineered then themselves to make it more effective, which basically, if you express this molecule in specific tissue or cells, it will biotinylate every protein that comes from this specific tissue. And they have used that here in tumor cells and implanted it into tumors in mice. And I keep that short, yeah. And you see on the left, on the right side here, that in these tumors, these teratomas in the mice, only the mice which really express the so-called CRE-positive mice on the right side, which express this biotin ligase, biotinylated all their proteins, which you can see then through these multiple bands that show up here in a so-called Western blot. And only in these mice, we find these bioattenuated proteins then in the plasma. And the beauty of this bioattenuation is anything that has biotin on it, you can pull down easily, you can grab and select and then analyze what is there by biochemical techniques. And so you can actually exactly see in the plasma which proteins are now released specifically from these cells or from this organ where you express this molecule. So with that, I hope I set the stage for scope of the problem. Yeah, you see interorgan communication is a major problem and a major driver for the development of multimorbidity, but it's really a very complex scenario with multiple different signals, with multiple dimensions, with relays, with various modifiers that work in. And it's so complex that we can't do it with an Excel spreadsheet. We need AI in order to analyze these data and in order really to understand, model, and prevent multimorbidity. Thank you very much for the attention.
Wolfgang, are there questions for you? Just to add a little bit, with the planned analysis in our excellence cluster proposal there, we will we can combine data from health record data, so health record data with molecular data describing these signals, quantifying these diverse sets of signals in well-characterized patient cohorts, and patient cohorts gathered in Berlin, but also at various places throughout the world where we can compare them. So there's really ample of data we accumulate, and are happy to collaborate on that aspect also, because this is a major undertaking, which we cannot only do with classical wet lab-based approaches. We need, as Wolfgang said, new tools to identify the most informative signals, the signals driving the pathomechanism, which we can then use to develop new therapeutic approaches. Yeah, that's the idea, and we do that with the data we have accumulated already, and we will prospectively accumulate throughout the cluster in just prospective observational studies, but also interventional studies. And so there's a lot of data. Questions?
So this is based on historical data that you're making sense of?
Part.
Yeah.
So there is a lot of, any data basically we can get our hands on. There is a lot of existing cohorts, healthy cohorts, single organ disease cohorts, as well as multimorbidity cohorts, on which we already have biosamples and good clinical records in phenotyping. We also have ongoing cohorts that we want to follow up longitudinally, and we're working with a lot of international partners who have health data records internationally, who have biobanks, in order to really also see the global dimension of this problem in different populations.
But also, in addition, we have, as I said, interventional study cohorts. Just an example, the an inhibitor given to CKD patients, or patients with chronic kidney disease, is now known for its dramatic preventive effect of cardiovascular diseases. And there we have interventional trials where we gather molecular data more in order to understand why therapy given to patients suffering from a kidney disease is preventing cardiovascular disease. So it's not only observational trials, so following up the development of a secondary organ injury, but also really interventional trials, which can be really informative. Because
where you have basically gain or loss of function of individual organs and can see how that affects signals and other organs. Another example, of course, is, for example, kidney transplant, yeah, where you can see in a patient receiving a kidney transplantation, how that affects all the other organs, yeah, and hopefully for a betterment, but also the patient who donates the kidney, yeah, and suddenly only has one kidney left rather than two kidneys, which is generally considered not to affect health and don't want anybody to prevent from donating a kidney if you have a chance to. But we don't understand how that affects communication between organs and function, yeah, and that's of course a unique scenario really to look at in a clinically relevant human scenario.
Yeah, I'm interested in endocrine project.
Yeah.
And I'm interested in future biometric sensing. So being able to read hormone levels longitudinally in real time, down to CGM.
Yeah.
So it's interesting to potentially in the future combine those two data sources.
Absolutely.
Historical data.
Talk to me about that.
Absolutely, and that's exactly one of the challenges where I mean, I'm a physiologist, I'm not a big data person now, but where we look for input and help and collaborations.
Please hit me up later on that tech because I need to know about it.
Sure.
Yes.
Can I ask a rather naive and speculative question? So I'm from the UK National Institute for Health and Care Research, from the Global Health Research Program. So I was thinking about this notion of insults, right? Whether it's aging as a process, but in populations that might experience other types of insults earlier on, whether that's early infection, whether it's nutrition, whether it's climate insults, would you see that as other potential modifiers that might make those populations more vulnerable?
Because that's where we have the most data on right now, but exactly correct, yeah. Diet, of course, is a major modifier. Prematurity, of course, for example, or anything that happens actually in utero already. We understand very little about it. We start to understand how it affects single organ disease. We don't understand at all how it affects inter-organ communication and multimorbidity at this stage. Environmental factors, pollution, not really addressed in terms of multimorbidity and inter-organ communication. Yeah, so all of these are very important factors. Problem here is that most of the current cohorts and studies don't really assess these factors. So we don't have a lot of data available on those, but that's exactly what we aim for in all the cohorts and studies that are coming up. Yeah, also psychological stress, mental health factors, same thing. Yeah.
And we actually reach out through one of our investigators to other areas in the world, so in Africa, where other modifiers are operational, and therefore also other types of multimorbidity and pathomechanisms are operational, so that the problem is still existing, but it's maybe different, and would be good to compare, undergo these or perform these comparisons in order to understand whether there are similarities or differences.
And
of course, of HIV disease and diabetes, for example.
And there is a sort of little seeming contradiction almost that in lower income countries, the incidence of multimorbidity is generally reported lower, but that's
very incomplete data, though.
Exactly. Yeah. That's probably incomplete data, and of course, again, age, is a major factor. So it's, I think you have to be very careful in making these comparisons. Yeah, but there's a lot of very important information to learn.
Just a note, we're going to do a gestational diabetes cohort in South Africa, for example, and compare to a Swedish one with some very interesting possibilities. Precisely that.
Yes.
I have a question about accessing enough data. So what do you find is the biggest barrier there, that the rich enough data sets don't exist, that you can't find the right partners, or there are data privacy issues preventing you from accessing?
Part of everything, yeah. So, I mean, finding the right partners, of course, is work. We were fortunate over the last years that we've been working on this initiative to find really excellent, good partners in the UK, in Israel, in the US, of course, in Central Europe, and also in the global South to work with. Yeah, still more partners and more data would be better. Patient privacy, depending on with whom you work, and sharing samples for biological analysis is a challenge at times. Yeah, and that's a demand. And then, of course, getting complete data sets, because every data set is, of course, different and analyzes different things, filling the holes of what you don't have, and in the end, making sense of this huge amount of data, yeah, and bringing it together.
But that's why we also have a substantial part. They are focusing on this kind of data integration across different cohorts, and we also have one part focusing on developing a so-called data knowledge portal, which allows us within the consortium to better compare data and bring the data together, but also to share the gathered data with the community. So we know that there are a lot of hurdles, but there will be a substantial part really focusing on that one. Especially in Germany, we are lagging behind. We have to say with aspects about sharing data and also gathering complete data set from patients. Unfortunately, we only just established the idea of health record data in Germany, and that's why we also reached out to other countries. Yeah.
But providing a platform that would disseminate and make this available to broad community is exactly also what that would be.
Okay. Don't hesitate to ask further questions and reach out to us. We think this is an absolutely important topic and need to be addressed. And we are only, as Wolfgang said at the beginning, worldwide, and it's definitely the next pandemic. Also in the news is announced as the next pandemic.
Is it I can be about.
So we'll proceed with the next talk at 11:30. We'll have a break.
Yeah. Next time. Yeah.
Das sind von den
Yeah, it's emerging, not no violence. Absolutely.
Aber nicht Data.
Yeah, we have a very big initiative going on, which is quite advanced. But yeah, so there's no challenge.
How much AI
This
definition
is very relevant.
Also
Which one of the UN? That is a thing.
Ganz genau. Deswegen das ist ganz gut. Aber das ist eine der
Das ist voll die Idee, wir brauchen schon und
Yeah, I think this is like this spazieren an these pounds are
weil das was mir startet, ist Migration, also
Yeah, no, we should.
spatial transcriptomics und wenn sie daraus so eine Art ich weiß nicht, ob mal jemand
Yeah, I'm seeing him on this Thursday, so we find out where we are.
Ja, aber
Because it's all to do with the so it does make sense. So
anything with the ancestor
entwickeln können.
set up, we really
Genau das, was wir immer machen wollen.
the whole idea.
Aber nur Input und Daten.
Are you doing any sessions then, or is that, are you doing any sessions there? Private?
It's just literally research.
We're very much in a.
So you're around tomorrow. Tomorrow might be catch up.
Yes.
For evidence or to show what our business is.
But yes, we just use very map approach. So basically, we created these novel data structures to the noise preserve the underlying privacy. Well, never actually accepting any of the road by road and then you can transfer whatever size you're doing you want. Because the patient data never leaves the site. And it's not that clear. The only data structure that France or Spain, for example, said, oh, it's okay, you know, you're not actually sharing any of them. And it preserves all its files.
Thank you so much.
Yeah, there's studies like this.
Or you.
No.
Apply complicated.
Yeah, that's great.
Not just because you can do very advanced like type analysis. Yeah, many things. Yes, basically it's capture the statistics or equations the relationships between Thanks so much. 1 million rows. It's compressed to 8 rows, and then the width is basically the permutation of pairs of columns. But the specific statistics that we capture, it's enough to do most analysis that a researcher would want to do.
Yeah, and so if the individual patient doesn't show up, and so.
No, they don't exist anymore. I mean, but all of those relationships between features and, you know, how that whole
Language as well.
but it's been very, I'm from more data science and background, it's been very interesting for me to discover all of these issues that the healthcare community has.
Yeah. No. But that's, yeah.
Yes.
And that's very interesting. Do you have a
So I'm so sorry, I don't see hard, but
Yeah.
I have LinkedIn. My name is Ashley from
The next bit looks quite technical, doesn't it? Was it too that we thought it was more?
Yeah, that's, but this is a different organization. I have
Sure.
also work
Yeah.
for.
Like, oh my God, it's
Yes.
Yeah.
Yes.
Fine.
So like.
Yes, you can find me on LinkedIn. Yes.
They're like.
Yeah, because we're all currently
Eleven
building the same
till
for
11:15 on the ground floor.
kind of
doing that.
international registries or research databases for NASH, for example, for a bunch of different a bunch of different diseases, and it's so great that you can actually capture the analytical value while not even having to worry about regulations. Yeah.
When NHR launched the AI for multiple long-term conditions method, they funded the Alan Turing Institute to provide a research support service. Contacted them directly, yeah. I'm gonna make a note of those things, just link at you tomorrow in an email, just sort of link around.
They are not harmonized.
Very interesting. They're also
Harmonize the way the replications are phenotyped.
Yes. Yeah.
The diagnosis given by one physician is not identical to the diagnosis given by another. They use different terms.
And then I've stopped writing things down because I'm like, if I write them down in like, but you stay in my notes.
Because of the measurement technique, for example,
And the kids and whatever.
how do you adjust for this then? I mean, do you look at the distribution between different centers and then?
Well, I think
normalize?
in general, we try to do the analysis then related to this within one cohort and see whether we find and sort of to normalize it to these values within the individual cohorts. Yeah. Because it's so hard to compare between. I think some
Terms, which of course then also vary. Yes. You can try to adjust to the local references.
Yeah.
It also requires a lot of bioinformatical expertise.
Yes.
The example that I would just, I mean, for many things, it's pretty standard now. Yeah, but for example, renin levels, yeah, as a classical hormone, yeah. You can't compare between different hospitals. Yeah, it's always what I hear from our kidney experts. Might of course, is to say no. You have just a reference to the norm value in each individual case because it's so different.
That's, yeah, it's something we look at is, so when we create these data structures, you can then compare the distribution of columns or features between different centers without actually comparing their data, which is kind of cool, and then adjust based on this.
That's what you have to do.
Yeah.
Any of these vaccine?
No, we don't develop vaccines. For the time being, we only develop medicines.
Right.
Yeah.
No, to real it's awesome like that. Very too young as a
Everyone knows from HIV.
Yeah, actually, we collaborate. I mean, we don't develop the medicines now. We are like a development partnership. So we work with GSK, Novartis.
Across different centers, that also important in that time that they work very hard to
provide
everything else.
the discovery.
And that's why
All the discovery. So I do the pharmacometrics model,
reference rate for each
discovery, and we have the project management expertise.
So complicated.
Yeah.
But it's such a pity to think, well, I mean,
Did you
obviously
do
the quantity of data
trial
you need for these
optimization?
very
Yes.
complex.
Found lots of
Such a
trials.
pity to think of all this
Yeah.
valuable data stuck in silos or not properly labeled or normalized and
You
then
travel a lot. to-
the secrets remain.
So I do not specifically, but I must say I joined last December. No, I'm a physicist. I'm a biological physicist. So I'm doing a lot of modeling of cellular processes.
Yeah.
For joining and now I'm on the system level. Really interesting. So yeah, I didn't have the opportunity to travel a lot, but I definitely have some colleagues, some medical doctors, because we also take care of the
Kind of like a,
remote regions where the medicines have to arrive.
Yeah.
And so we need a lot of people that go and just communicate with them.
Yeah.
It's the fact that the medicine
Thank you.
they come out.
Yes, yeah.
And yeah, there are people that definitely more on planes than on the ground.
Yeah.
You're speaking Japanese, right?
Yeah.
Yeah, unfortunately, I have to leave. Oh, they're public?
Well, I think they're gonna make.
Okay, I didn't know. I was quite confused. The
Yeah.
website of the event was a bit
I know.
confused.
But they said it, it's not live streamed, I think.
That's great.
Next generation. Mein Sprecher wird Thomas Wiegand.
So it's kind of like, kind of cool.
I'm quite sure, you know, the audience
Also ich habe
is quite diverse.
It's really diverse, but
Yeah, I
the
know.
subject that really
Habe ich aber gestern gelernt, dass man
attracts many people.
I guess there's many people working on AI machine learning that are looking for applications, so I guess there's sort of.
Right, but did you have the impression that in the audience?
Ja,
this
aber
morning,
auf seinen
people
LinkedIn gibt es sozusagen dann nur
For instance, we don't use AI yet.
Yeah.
So I'm here mainly to see what people are doing with it, to see if it's integrated in our workflow.
I mean, same in my talk. There's no AI.
Okay. So, yeah.
It's more about,
Okay.
well, actually data source and how to conceptualize.
Ja, das ist nicht gut. Aber man darf jemanden.
Ja, aber da war das darauf.
Prospect many diseases, different
Mag sein,
diseases they share
mag sein darauf,
how to
aber
identify that.
Und gute Diskussion.
Wo man nur noch das Wasser.
Are you an epidemiologist? or-
Well, I'm a clinician.
You're a clinician.
Yeah. I'm internal medicine. My subspecialty is in clinical pharmacology. But my research is largely about using genomic approaches to identify and then validate.
Okay, I see.
Some clinical, some genetic epidemiology, and some.
Right. And you're not using any machine learning or AI to
Well,
pretend
people in my group, but largely the applications there so far have been in disease prediction. So measuring genomic, other routine clinical markers as exposure variables and using them to develop models to see who will get disease. Typically though, disease prediction is pretty tough. Because none of I mean, typically what we find is that any exposure variable that you measure, whether it's genomic or some other measure, is usually has a Gaussian distribution
Yeah.
and then a log linear relationship with risk. So most events occur in people with average levels of the biomarker.
Yeah.
They're at intermediate risk. They're California cases.
Yeah.
So then it's quite hard to separate people that will get disease based on the marker. And even multiple markers, when you add them, even if they're independent, you sort of gain a bit. But when you add markers, what you find is that the distributions, standard deviation broadens. So the separation, so you gain on the separation per unit areas.
Yeah.
So it's quite, prediction is quite.
Do you also do longitudinal models?
To some extent, yeah, because we work on longitudinal cohorts. So that's the prediction side, but actually most of my work is genomics to identify and validate.
Yeah.
Not prediction, causal analysis.
Well, yeah, which is at least equally important, I would say, because in this video, I mean, one of the things that I noticed when I changed field is that, I mean, in this medical, when you have those medical data, it's very hard to distinguish correlations.
Yeah.
Possession from correlations, and I think it has to be very careful that, you know,
So
interpreting the results.
No,
of this.
absolutely. So so this is why for that aspect, we work largely speak on genomic data. Because genomic associations are to some extent protected from non-causal relationships between the measure and the outcome. The reason is that usually in epidemiology, you measure something in the blood, higher in people who later develop the disease. Could be causal, could be reverse causation. Early disease you haven't yet measured and it's causing the change of the marker rather than the other way around, or confounding the marker. There's some other factor you haven't measured and you don't know about that causes the disease and the elevation in the marker.
Right.
But with genomics, it's protected because disease can't affect your genotype.
Yes.
So reverse causation.
How does this communication work?
And confounding is reduced because genetic variants are allocated at random. So you get randomized application of exposure. So most of the usual factors that affect standard observational studies should be balanced between genotype groups. So it's a bit like doing a natural randomized trial.
I see.
So we use the kind of randomization to
higher
gain, I guess, we would hope a more confident just causal
on average.
Yes.
You can start tailoring.
That's the handy.
Yeah.
Something that I think about is how much we can actually understand from the genes, in the sense that true they are upstream many things. Then
Good example.
we find, you know, some correlations between a specific genotype and a disease. Does it really mean that, you know, if you have that gene, you are first to get the disease, or because ultimately there are pathways that are intermediate from, you know,
no, totally. So the effect sizes are really small. They alter the probabilities. For most probabilities. But if the associations are real, in other words, it's going to demonstrate them on a substantial scale. Then you know the protein is affected. Then because proteins are targets of those drugs, you can generally develop a drug that's much more potent.
Als Template, aber wir haben ja schon
you know which protein to target. So that's the kind of so what we've done is another technologies is done many kind of
Ich wollte das jetzt aber nicht nochmal
known drug targets. So we know and then we look at genetic variants in the gene that encode those targets and see if we can reproduce them.
I see.
And then, having shown that, then you say it's a discovery. Yeah, so mostly it's around that contradiction. From what I see, mostly that is being applied in healthcare sector.
Und das eigentlich arbeiten.
Prediction on the one hand, the other is kind of
Yeah, that's
and I think it's pretty good at that.
yeah. Apparently, at least from the publications that I read about it, it seems like. And especially if they are combined, like if the image recognition from the machine is combined with the expert knowledge, you know, so there is both a human side as well that validates.
Exactly. And takes responsibility for that.
Yeah, because unfortunately, that's what the machines cannot do. They cannot sign in the end, you know, and somebody has to take the responsibility.
Wahrscheinlich mechanisch.
The other area, I mean, in imaging as well, is to kind of rapidly quantify image features. So in UK,
Inhaltliche Aspekte, dass man auch wirklich nochmal sagt, inhaltliche Aspekte achten wie Organachse, Austauschbeschreibung, Modifier.
Das stimmt alles.
Das sind die Ideen, which of
So the data's available
Huge
for use, but it's huge data. And so we're particularly
das könnte
interested,
man
for example,
eigentlich
cardiac.
so
So the heart has 4 chambers, have a
formelle Vorgabe machen, dass man sagt,
have different
Integration
thicknesses,
Expertise.
points,
Yeah.
relevant, chambers have different shapes.
Weißt du,
So
dass das irgendwie
we've been using AI.
auch
People in the
visualisiert.
P4X
Genau, man könnte,
rapidly, going through
man könnte
hundreds
das
of
als
Yeah.
MRIs and then producing
Checkbox machen, ja?
quantitative
Communication
rates based on those.
So without having any
Data science,
suggestion as to what to look at, like the AI determining what was interesting to look at, so like
Yeah,
yeah,
and
supervised.
you can even, but you can also specify, so for example, you know.
Es erscheint dann nicht,
So we've got measurements. That's proved helpful, because then you can combine it with genomic.
Aber das kann man sich dann überlegen, ja, ob, also dass du sehr asks.
No, it is great, yeah, these are great.
I did, I'm so interested partly in using these things called polygenic risk scores, where you sum the risk variance in a gene in an individual, some information about that risk.
Es ist die Entwicklung der Highlights, um wirklich zu sehen, welche
In this field.
Dimensionen, wenn ich es jetzt mal
I was interested to know how many companies there were
und
working
eben
in
nicht
this
nur,
field, what they reported.
sorry, Mitch untersucht,
But
TT.
most of this information is not published as a research paper, it's in the web, you know, it's in documents.
Yeah.
So my own personal foray to AI is to use ChatGPT, ask the question,
Yeah.
can you tell me how many
Ich weiß ja mittlerweile, dass man
doing various
Risiko mit 13
It's sort of helpful in a way.
So GPT doesn't have access to the internet, right? So it's not, well, it had until 21, yeah.
Nee, bis
Ja.
die 10 Jahren, die Analyse hört ja in 10 Jahren.
Ja, aber die hast du falsch kommuniziert. Das sagt man nicht.
Klar,
Yeah. Unfortunately.
Are there any more? Yeah.
To satisfy your special function, that's the problem.
Auch eine Lungenentzündung.
And did you check that the names were actually
So now I've
corresponding
got
to
this big
real,
spreadsheet.
to actual?
So now I have to do
existing
some manual
companies?
curation to see whether.
Yeah,
it's
because I had to check that it didn't come up with, you know, invented company names, which is something that those language models, unfortunately. I use it to, well, when it first came out, I tested its understanding, well, it doesn't understand anything, but it's semi-understanding of physics, and it was pretty bad. But then at ChatGPT, well, like, you know, if you were asking questions, you know, I have a ball, a glass, then I take the glass, reverse it, then I turn it back on its original position. It is all happening in a table. Where is the ball? It would usually answer, oh, it's in the glass, because that's, you know, right?
Yeah.
So, but now GPT-4 is better at that. It can actually understand that the ball went into it. It has some, I don't know exactly how they managed to train it.
Du kennst ja meine
Physical
Version.
understanding, but it knows that the ball.
So you're saying it doesn't have access to the web?
I don't think so.
So how is it doing?
What it's doing in what sense? Like the research.
And how is it making predictions?
Ja, ist auch irgendwie auch kein Mensch.
Well, because it has been trained on content that is on the internet, without necessarily having the consent of the people who produce the content, right?
So you're saying it had, but now it doesn't.
They probably fed it with all the whole Wikipedia, you know, pages and whatever that they found. But it doesn't have current, like it's not, yeah, it doesn't have online access.
Yes.
to internet. There are some plugins that you can use, but I don't know very much about it because I don't know the program.
Same issue.
Yeah, I guess they're all pretty much the same. I don't know much about the other. Well, we like there is Microsoft Copilot, which is kind of a good. I don't know. Yeah. But I think they all. I mean, the concept is the same behind all these machines, and it's it's not that there is anyone.
So I'm gonna talk from.
Das ist eine genetische Veränderung.
UCL, because he did his PhD in UCL.
Durch diese klinische Hilfe
He was talking
da gibt's noch überhaupt keine
Und die heute
interesting thought. And now what there's.
Doch, das ist gar nicht so.
So then we
Isomorphic, yeah, sure, yeah. Yeah, which is fine. I think the product is AlphaFold. Protein folding, which was, I think, one of the most famous successes of AI
So
in
we
recent
will
years.
continue in about 15 minutes and
really
touch upon different
Although,
aspects.
I know people working on protein folding from the non-AI, and they're still skeptical about, like, in the sense that sure, it predicts to a fair accuracy the
ex vivo model systems.
quality of these proteins, but then can we say we actually understand what is going on, which is part of this whole, interpretable machine learning, like we get the right answer, but have we learned anything?
Different organ systems.
So it's good, to some extent it's good to make predictions in the sense that you trust the result, you have checked that the result is correct. But as long as you do not understand how the result is obtained,
we cannot
trust. Like
retrieve all the data which is necessary from patient samples, especially.
We are trying to implement. We have this idea of implementing AI in pharmacological modeling. Because sometimes we, especially for malaria treatments, we do not like to give single monotherapies because of drug resistance development, right? So we prefer combos. And when you do the modeling of these combos, you often have drug interactions that are complicated. Like we don't exactly know how they interact mechanistically. So then there is a question. We know something about the dynamics of these drugs, and we know, for the individual drug, we know how it is absorbed, how it is eliminated, but then we don't know exactly what happens
according to the sex
the body in terms of the interaction. So can we train a neural network to learn
approaches? We completely
these interaction from the longitudinal data
give
that
the
we
same
have,
dose,
clinical
the same
trials?
type of medication.
That's something that we
Everyone knows that it's wrong, but it's not considered at the moment.
to see whether others are doing anything like this.
I have another talk which deals a little bit about.
And that's an area that's quite interesting, I think, from my perspective, because
No,
it's a situation where you don't just
in 30 minutes we will
take a
start
neural
with the
network
next
that
talk,
has
and
no,
then
that
go
is,
2 talks, and then
say,
we have another
ignorant,
break.
doesn't have any domain knowledge, and so it is free to learn unconstrained, but you actually take a model that you already know something about, so you have those equations that describe the pharmacokinetics, some elements of these equations. And so you fix this part of the model, but then you leave a part that is the unknown part, and only that part you ask the machine to learn what is actually going on. So this is a kind of constrained learning. Some people call it scientific machine learning, but it's bounded by the domain knowledge that you have. And I think it gives. I'm more prone to believe, the predictions of this hybrid.
That's interesting idea.
In this talk, by
Ende ist
describes the sort of early development.
Für mich nicht. Für mich ist das Ende 22. August.
Warum?
Obviously, they worked on.
Da ist die offizielle Einrichtung, Einreichung, aber es muss wann mal genau sein?
Mitte Juli, da gibt es keine
Game explained why it's
ehrlich gesagt, ist mir vielleicht egal, wenn ich zufrieden bin am 15. Juli, dann müssen wir weiterarbeiten.
And so they trained
Nur Stempel küssen.
AlphaGo, so neural network, to learn they gave it the rules,
Der
and then
politische
put no
Simulator,
more,
dann
and just said
Yeah.
learn.
Mhm.
And then essentially the ways they get their systems to learn is that they
Aha.
make a system that has
Das ist
knowledge,
Urlaub.
and then they develop another system. And they
Sie wollte es haben, weil sie
Jetzt, es ist so ein spannendes Konflikt. Wenn du dich da irgendwie.
way. So anyway, they
Ich bin ja,
Ich habe eben jetzt schon Leute geredet, weil sie haben gesagt, ja, er hat 2
they're
Wochen
going to
lang.
challenge the world's best Go player. And they did it, and it was all televised. So
Okay, das ist auch
the machine makes this
spannend.
crazy move.
Crazy moves, yeah, I remember reading in the news about
Yeah, so I wonder whether.
the Go Master was pretty shocked at the move of the ultimate
And then
team.
apparently it's all rewritten the whole concept.
Yeah, also because it's a game where I think there is a very strong aesthetical component to it.
Yeah.
And apparently the master player
It was
was
an ugly move.
exactly.
Yeah. So yeah, I guess this balance between constraining
Oder
and
verstehen,
leaving things unconstrained.
Signale gegenseitig beeinflussen. Es wird ja am Kopf
It's a good one. I guess you need to be confident. What you think you know, you are confident. in what
Yes,
you know.
absolutely.
Ich habe jetzt gerade so einen Stream gedacht.
But I think it's still, I mean, to a fair extent, those pharmacokinetic, there are some parts of these pharmacokinetic models that we have a lot of information about. Some parts are easy to measure. I mean, in terms of the, you know, rate of elimination and rate of absorption, these we can measure, so.
Yeah, and I think
Aber da haben wir
we broadly understand it from, you
Ja,
know,
ich
drug
weiß.
development.
Aber
Is that probably 3 or 4 decades ago, a major reason for drug
Nee,
development.
kann man letztes Mal. Das hat so
Yes.
Tools, wo man so.
And that then got solved
eigentlich ganz was machen kann.
Yeah, because of the mathematical models that
Yeah.
were not, if I understand correctly, I'm still not entirely expert of the history of the field, but like those mathematical models were not
wir wissen
used to
mittlerweile
make
schon bei diesem Zellplan-
predictions, they were more used just to describe what was going on.
Yeah.
China
Which
gegenseitig
is a
negativ
bit different from
beeinflussen.
what we do now, because now we
Was sie
get data from, you know, a phase 1 trial.
Yeah,
Musst du
and
mal
we
so eine
try to predict what
T-Zelle oder eine
happens
Immunzelle
in the phase 2
zu China.
yeah,
before planning, not to actually optimize the
Yeah.
data.
Kriegt aber gleichzeitig auch
But in
noch
our NGO, what we do is a lot of
all die drei, das sind komplett
preclinical to clinical, you know, we have the data in vitro, can we do a first in human
das
dose
sind aber
prediction
ähnliche
from the in vitro data, or
gleiche
um,
Komponenten wie der
which is really important
Signalkapazität.
for malaria, we get data from phase 1 studies in adults.
Ja, müssen wir einfach
And we
mal
translate these
etwa
into
96er Platte mit
predictions
T-Zellen
for
drauf und
children.
dann alles
Ja.
in Kombination, was du dir
Which is
vorstellst.
also something that
Und
is
gucken, was sie machen.
quite challenging, and I
Yeah.
mean, we,
Und sowas kann man aber easy und
yeah.
wenn du-
So now the major reason for
wenn du
failure
richtig
is
häufig
you
mit
chose
diesem
the
Screen
wrong target.
koppelt und mit
Yes.
häufig sammelt, und da hast du doch Daten,
Ja.
da kannst du dich deine Leute dann aussuchen lassen.
Sorry.
so your UCL,
Ja, ja.
it's a
Yeah.
lovely place to work.
Ist da drin
I risked going there for my postdoc actually
Das ist
after my
schon die
PhD.
erste
Right,
I got an offer from a group that was there at the time, but
Yeah.
not there anymore, and from Geneva, and then I ultimately decided to come to Geneva. But
yeah.
yeah, no, I was very
It's
impressed.
it's very big,
Yes.
actually, and even if you work there for many years, you don't quite know everything
Yeah.
that's happening. So,
And for me, I mean, coming from biological physics, it was quite an exciting place because of the proximity to the Greek Institute,
Yeah,
you know, and
yeah.
the potential collaborations with
yeah,
us.
that is very good, that's a good.
Yeah.
And actually, there's quite a lot of collaboration across London.
Yeah. If it weren't for the cost of living of these
Oh,
cities, it would be great for science.
I know.
I did my PhD in Paris, so I was sort of used to, you know,
Yeah.
the big city with many universities,
Yes.
but sort of, yeah.
Yeah.
She's been there.
C'est plus clair, ça va mieux.
And I also think since Brexit, it's become less attractive. It's difficult now to point people.
It is, yeah, and it was maybe even more the case when I was interviewed at UCL, because it was really, it was 2019.
Yeah,
So it was still, you know,
lots of things happen.
lots of things, exactly, you know, still unknown, and yeah, it was actually part of my decision actually not to go, because I didn't know exactly how it was going to,
Yeah.
the situation was going to evolve.
Oh, okay.
Now at least we know Horizon.
Exactly, because at that time with exactly, I knew also that I could not apply for, Marie Curie fellowships or, ERC grants. Now it's different. Now Switzerland has this problem.
Oh, really?
Yeah, they're not part of it. No, they haven't been for, I don't remember exactly when they stopped being part of Horizon. It wasn't long ago, actually. So there have been a lot of protests, of course, and now they are trying to go back to the Horizon program, but for the time being, the SNSF is replacing those, like ERC grants and Curie fellowships with, national owned
You can go back to the same during this.
Right. I wonder if it actually costs less than being part of the Horizon program.
It may cost less, but I guess
They actually lose competitiveness, right? So it's, for instance, when I didn't get one of these fellowships, but I tried to apply for one of these SNF fellowships, and I had the problem that my project, there was no commission that was, for biological physics. There was no commission I could send it to, know that the reviewers would be experts of the field.
Yeah.
Which wouldn't have happened if it was, like an application for a Curie fellowship. Exactly, you have a smaller pool, so fields that do not exist in Switzerland but do exist, in Germany and France.
Yeah, and they don't seek international review.
They do if you pass the first phase of review.
But you need it for the triage.
Yes, exactly.
Hat jetzt recherchiert, also Depressionen haben ein höheres Risiko für
Dialysepatienten mit
Depressionen.
Yes.
They're now implementing.
Impact-Faktor? 2,3.
Wie oft zitiert H-Faktor?
Ich habe mal die neue Sachen, wo du große Datenmengen brauchst.
What was this issue with Northern Ireland solved actually?
Also, so
Ist sie nicht jetzt irgendwie Abwehrsynologie? Nicht Abwehrsynologie.
Factory solved. No, there are still checks. So what they politically is difficult to say that there should be checkings between mainland and country.
Okay.
Yeah. So, but they don't really know.
Ich schreibe mal was.
Goods that are going to Northern Ireland.
Man muss sich ja nicht auf Dialyse so schon einstellen.
Ja, Dialyse ist halt total sondern es ist nicht
Muss ja nicht sein.
automatische
I see.
Ja, aber
Yeah.
You have the elections, right? So maybe who knows how the situation is going to evolve.
Parties that are
so ist das Bauern. Weil dann selbst ist das dann auch
Die Einschlusskriterien einfacher.
Genau, du hast ja 1000 Dialysepatienten.
I am so sorry.
Das kann halt auch schneller annähern.
Genau,
Die anderen sind niedergelassen.
Genau, es ist halt highly feasible for us.
Reverse.
Yeah, maybe
Is it
just at
possible,
least have a
like,
customs union.
Okay.
Not to have freedom. Who knows?
Good luck with that.
Yeah.
Yeah.
I'm not holding my breath.
ist ja schon ein Wort, da sagt man heutzutage was an.
Die erste, ja, sehr generell.
There was someone it might be free.
Deswegen gebe ich ja jeden gesagt, extra nochmal Preis. Aber die Fullscreen.
Das macht es halt mal.
Seine eigenen Images.
Ja, das ist sogar selber gebaut.
das weiß ich aber nicht, ob die Immunprozess nach.
Ja, aus dem Fenster, aber das haben wir in der Pathologie. Ist nicht schön.
Ich finde es sehr schön. Nee?
Das war Titel bei der Multimorbidity. Ziemlich viel Zeit reingesteckt.
Ich habe es noch nicht auf dem Kanal geschafft.
Ja, das ist auch eine andere Zeitschrift.
Aber zu langsam muss man wirklich so ganz
Dass wir in einem angenommenen Paper jetzt über NCBI, NIH, das irgendwie reviewen, verifizieren und sonst was muss, damit es ein DMC und sowas kriegt.
Ja.
Ja? Das ist ja echt total painful.
Das ist ein großes Problem.
What is about deciphering networks versus between in the twin? I've already identified. So they were running the tests on against it.
Also Martin hat jetzt ein kurzes hier mit Bruno hier.
Okay.
Man muss den Leuten immer nur sagen, ja, mach's doch. Also wir haben jetzt nur die ganze Zeit gefragt, Leute. Machen Sie mal das.
Was there anybody
Ich schreibe jetzt ganz
heard on the Solomon team?
Mach das schon.
Also dann
Ich dachte, wenn ich selber machen muss, nicht so viel Fleisch brauchen.
Ja, also
war nicht gleich. Okay, die werden mehr viel
Okay, so welcome back everybody, and it's really cool that you came back from the coffee break. We will now switch a little bit gear because we also incorporate certain aspects to our proposal which are not directly data from patients, but relates to the fact that you, in order to get massive data, especially longitudinal data, you need to perform some in vitro analysis using human model systems, which allow you to also model complex signaling processes and interactions. And that is what Niels Blütgen, professor of computational modeling at the Charité, will talk about and will embed into our proposal.
Okay, hello. Thanks very much for inviting me to this workshop. So really, I will now concentrate on a rather specific problem in analyzing multimorbidity and also inter-organ communication. This is really looking at the receiver side, looking at the cell that processes signals, how we can influence that, what mutations may cause through this, and so forth. And so this is really more detailed mechanistic understanding, which so far has been really hampered by mainly the experimental approaches that we have at hand. And so the data sets are relatively small that we can generate for this. And so what we currently have been doing, but we're switching now to other methods, is really looking at more mechanistic studies, more mechanistic models, small scale models. And now the experimental methods really change, and I show that at the end, that really give rise to data that we can use also to have larger, more AI type of. So currently it's more deep thinking, and then we move to deep learning in relatively short time, I think, and the community moves there. So our group specifically focus on signaling that is involved in cancer. And as many of you may know, cancer is really driven by mutations in signaling proteins that actually relay growth signals from the outside of the cell to the nucleus. And many of the components of these signaling pathways are mutated and then constitutively signal, and the cell constantly receives signals that drives the cell into the cancer phenotype. And so what our group is actually was very interested in is like, can we actually understand when we have this network, which we know now for decades, if we just interfere with this network, can we actually predict what will happen to this network and also to the cell phenotype? And what will happen if we add additional oncogenes to these network, genes that drive additional signals or any other perturbation? Can we really predict that? And if the network would look like that, right? So this is the famous hallmarks of cancer map. This is basically reflects what we knew 30 years ago. Also still the thinking of many people in oncology. If the map would look like this, you have a signal that drives proliferation, you just block it and then cells should be like stopping that, right? But the reality is obviously more difficult. First of all, quantitative details matter. It doesn't matter whether there's a signal, it matters how much of the signal, when the signal, what's the frequency of signals and so on. And the other thing is really, it looks more like this, right? So this what we have as a concept of signaling pathway is actually a complicated network. We have a lot of feedbacks that we know also from engineered systems, for example. And we have crosstalks between all these pathways. So if you interfere with one of these pathways, other pathways will.
The center stage of the AI for Good Summit has started again. Please go to room D or room A if you have a leader's pass to check out the next round of keynote presentations on the center stage of the AI for Good Global Summit. Thank you.
Thanks for staying. Okay, so and then what's also another important problem, particularly when you want to design drugs for oncology, is that not only are these networks different between different patients yet.
Oh, just a small question. Yes. The connections are these correlation between the.
So this is what I have here is really protein interactions, but that's an example of so it's not only that every patient has a slightly different network, it's also that every cell has a different network. So we have heterogeneity within tumors and across tumors. And so that makes it actually a pretty hard task to actually predict what happens when you interfere with this network. And so what we do in the group and also in the community is really use model systems. This has for large, cell lines derived from tumors, perturbs them, use quantitative assays to assess how these cells respond to these perturbations, increasingly use high throughput assays, for example, transcriptomics or other high throughput methods to assess really the molecular composition after perturbation and compare that to before the computation. If you do this very systematically, you can then use mechanistic mathematical models to try to integrate that data and get mechanistic interpretation of that data. We can use it as well as statistical models to try to make sense of this data and also classical bioinformatics approaches. And so I just want to show you like one example of how we did or how we do this. We have a panel of cell lines that we can manipulate in the lab. We then make as many perturbations as we can by using drugs or genetic manipulations. Then measure how the cells respond to that using something like measuring, for example, phosphoproteins that are carriers of signals, and then use this data to calibrate a network model. And then, of course, the network model is something that we can analyze and make predictions. As what will turn out is like if you have these network models that map these data on networks, it is not only good for like making automatic predictions, it's also good to rationalize the data. So it's not only that you make automatic predictions, but you can also think about what would be good interventions, make prediction of good intervention into this network, which may, for example, stop growth, and then test this in more complex and more expensive model systems. So the area where we work on is a signal network that is one of the most mutated network in tumors. This is MAP kinase signaling that basically drives most of the solid tumors. And it looks like this is a relatively small network of interacting proteins. But what you can see is there are feedbacks, things crosstalk, and so forth. And so what we do is we perturb this network with drugs, with also adding, for example, extracellular ligands, and make a perturbation response map that you can see on the right, where you see basically the cell responds to this perturbation by upregulating one of the proteins. And then you do this across many different cell lines. And when you do this, you can see that these cell lines react differently to these perturbations. And then you try to integrate this into network models. And so for the networks that we are working with, this is possibly the most overstudied molecular network in mammalian biology. We know really the players, we know who talks to whom, and so we can really map out that network pretty well. Also because of the importance in basically driving most of the solid tumors. And so what we do is take this network model as a scaffold and then try to estimate parameters, which are something like, if you want correlation, it's not exactly correlation, but estimate parameters in these networks, remove edges where we think it's not supported by data, which are not important, basically statistically not important to actually explain the data, and then prune the network to a core network that really describes our data best. And then you can basically from these maps can look at the parameters. And for people that are not really in the field, maybe not so intuitive to look at these maps. But one thing that we noted is that one of the most important parameters in terms of quantitative strength is not really signal that flows along the signaling pathways, but it's really feedbacks where, as you see here in yellow, one of the last signaling pathways really inhibits upstream components. And this is something that after we saw this, like many people see this now in cancer-related signaling pathways, it is really one of the last components in signaling pathway inhibiting upstream. And this has very dramatic consequences when you start to think about therapies. For example, when you inhibit now within this pathway, your signal doesn't actually reach these components called ERK. What happens, you lose the repression of this receptor. The cell becomes very sensitive suddenly to external signals. And what happens then is that it activates also other pathways. That is, branches out, you have activation of other pathways. And that's one of the reasons why inhibiting in these central pathways was actually relatively unsuccessful in many tumors, unless you give an additional drug that inhibits also the upstream components. And that's can show this also experimentally. So if you give drugs that inhibit downstream pathways and upstream pathways alone, these are growth curves of cell lines and xenografts. See, they have no or mild effects. These are the blue and the green line. But if you combine them, you basically get rid of cell growth. Yeah? And that's why currently we see in at least for targeted therapy, targeting signaling, in solid tumors, you see usually a combination of drugs that target upstream and downstream. So that's kind of the current state of the art of therapies. So I was starting with saying, okay, these things are terribly complicated and super heterogeneous. And that's actually not reflecting somewhat in the type of data that we generate currently when we use cell lines. These are very homogeneous clonal cells that basically that don't really reflect the heterogeneity that we see. Our group specifically is working on colon cancer, and that derives from the intestinal epithelium. And that's a very heterogeneous and structured tissue. So you have basically a layer of epithelial cells that are stem cells and then differentiate and turn over within 10 days in your normal epithelium. They renew your gut every 10 days. And so you have basically different cell types in this epithelial tissue. And that's something with modern experimental systems we can also mimic. We can grow organoids, which are basically mini guts, which basically display exactly the same cell type composition. And we can do this from normal tissue. We can also do this from cancer tissue, which also is much more heterogeneous and reflects much better the heterogeneity in the tumor. So we ask very simple questions like, if you now add to the normal epithelium oncogenes that drive the pathway that we are interested in, this MAP kinase signaling pathway, it's just like the pathway that is mutated in about 70% of all those tumors. Now, what happens if you simply do that? And what you see is the red one is one of the oncogenes that you find in 50% of the tumors, and the yellow one is oncogene that you find in 20% of the tumors. You just split them into the oncogenes if you want, by transgenic approaches. And when you do this, you see something very weird. See, when you stick one of the yellow one in, this BRAF, unfortunately the pointer doesn't work, so I cannot point to that. If you look at the lower column, where is BRAF, you say then all of them light up. They all activate the signaling pathway. And when you look at these organelles down there, they completely lose structure. When you just go one step up in the pathway, it's a completely different picture. Basically, only very few cells actually activate the pathway, although many of them actually express the oncogene, and they retain the structure. So it's like, not that if you activate one pathway by an oncogene, you have always the same effect. You have very different effects depending on where you do it. And so we thought, okay, maybe we can use modeling to dissect that, to understand what restricts the action of one of these oncogenes. And so if you want to do this in heterogeneous tissue or heterogeneous models, so far it was actually very difficult to do those experiments because all the experimental assays required rather high amount of material. But in the last 5 years, we really saw a revolution in types of assays that we can do where we can measure molecular details really on the single cell level. And so what we do is combine these complex systems, the complex organelle system with perturbation, and then go into single cell measurement and try to understand on the single cell level how they respond. So the idea is of our organelles, make them into single cells for measurement, organelles with perturbation, and then sort them by what type of cell they actually are, and start to compare them and make models between these perturbed subpopulations. So you can do this, you can identify different cell populations here labeled with these different numbers, and then look at the molecular details of these responses. So I don't want to go into much detail here. And when you do this, this is basically how the signaling responds to different types of perturbation in different clusters, and compare the right-hand side, which is where you have an additional oncogene, with the left-hand side where you see, well, there's just a control. It has an epithelial organelle that has no oncogene. You see relatively little minor details. So you see mainly detail difference in cluster 6 and cluster 1 if you look very closely. But then we can actually ask, okay, why is that? Why is the oncogene that drives signaling in tumors not active, not activating signaling in cluster 3 and 4? And again, you can use models to dissect that, integrate this data into models, and ask basically which of the edges has a different weight in the different clusters. And so what we figured out is that it's actually important. There are 2 important points. This is, I think, maybe too much detail here, but you can use this really to mechanistically pinpoint to the parts of signaling that are actually relevant, for example, for restricting actions of oncogenes and then causing different responses to different oncogenes. Okay. So far, as I said in the beginning, the work that we but others in the community really try to train models on perturbation data, and what we use is basically simple linear network modeling, models that represent the data best and trying to make sense out of it, rather by analyzing the model very carefully by hand, thinking about it and so on. And so the major bottleneck so far was really the amount of data that we can generate, the amount of perturbations that we can do in one experiment in a comparable way to build really nonlinear statistical machine learning or deep learning models So that would require more perturbation at larger data set. And this is something that really developed over the last, let's say, 2 or 3 years, that we can actually do that experimentally. And so the main idea of what people, main developments that made this now possible is really genome editing, CRISPR-Cas9, where you can basically knock out genes in a really high throughput way. You can knock out many different genes at the same time. And do this really, really robustly, and combine that with methods that allow us to look at single cells in a relatively deep molecular phenotyping way. And if you combine this, then you can run experiments where you can knock out hundreds or thousands of genes at the same time, and then identify the effect that has on molecular networks in one experiment. By a number of tricks. So this is now possible. This is like really a field that explodes in the moment. And so by different types of CRISPR-Cas systems, you can also address different molecular questions. So for example, you can really do mutations, you can inhibit transcription translation, or you can also inhibit the mRNA at the end. You can also use small molecules and combine this with small molecules and inhibit certain things or pathways. You can really perturb at different levels of the molecular machinery of the cell. And then on the other hand, you can read out different levels of molecular machinery. You can read the transcriptome, you can read what happens on the epigenome, for example, and others with these methods. So there are now approximately 50 of such data sets where people basically knocked out large number of genes and tried to figure out what's the response of the cell to that. It's an enormous resource to train larger models, opportunities for AI. And so far, there was really one bottleneck. If you want to try to really work on this data, there was no community standard whatsoever. You just download the data, it was completely different analyzed. The gene in one data set was completely called by a different name than the other. So you really have to normalize this data, both and in the level of annotation, but also on the level of the data of the data. So it basically was useless so far. So I'm really happy that one of my PhD students took the way to really download the data, annotated all manually. It was a huge effort together with the Chris Sander group from the Harvard Medical School. And so we basically compiled this all now in one resource, scPerturb, which is actually taken up by the community. We have now 12,000 downloads, I saw. So it's really people use that, try to benchmark their methods, try to come up with models that can be actually predictive in future. And I think this is really the sort of data that will help us in the future to train larger models that become predictive and will allow us to predict what certain, for example, therapy will do on the level of a cell. So just to show you a little bit, look a bit in this data, what you can do is, for example, look at what's the difference between a population of cells that is unperturbed and has a specific perturbation, and then we need to think about metrics that allow you to define the distance. That would be one example. This is called e-distance, and if you do this on different data sets, so what you see here is basically on the left-hand side, on the horizontal axis, you see basically e-distance to unperturbed, and you see how strong certain knockouts perturb the transcriptome compared to the unperturbed. And you see that you have some things that really strongly affect cells, others affect cells mildly, and you can basically order things. What you can also do is compare different perturbations to each other and the effect they have on the transcriptome. And by that, you can, by just clustering this data, you can basically redefine signaling pathways as we did here. So you can really say, okay, this is signaling path. These are molecules that have the same function. These are molecules that have different function. These are grouped together and so forth. So really explore this data now. So we use these approaches now a lot, not only to look at the effects of loss of function of certain genes, where you inhibit stuff, but also what the effects and classification of oncogenes. So the idea that we have is basically put in oncogenes in cells and then look at what they do on the level of molecular phenotypes, and then try to start to understand whether different oncogenes have different phenotypes or classify oncogenes basically based on their phenotypes. So this is an example. For example, we do have like 50 different oncogenic mutations on the what you see here is, and this is the distance. So how different are they in terms of phenotypes? And when you do this in 2 different cells, what you get is basically 2 different answers. It really depends on what are the other mutations in the network, what your specific oncogene elicits. And with this, I think I come to the end. So what I wanted you to take home is really that when you train models on perturbations in cells, you can really understand a bit better how molecular networks respond to therapies. You can understand resistance to target therapies. There are now really a number of new experimental approaches that, for example, the combination of genome editing and single-cell sequencing that really allow to generate these large-scale perturbation experiments, which are really a big resource now, and where we will see a lot of development of also computational methods that allow us to derive predictive models. So, but we need these computational approaches to analyze these high-dimensional data, and it's really an opportunity, I think, for the AI community. And as I said, we compiled those data which are publicly available now in a normalized way in a resource which everybody is now open to use, and I hope we see really a lot of advances in it. Yes. Well, thank you.
Thank you very much. We have some time for questions.
Hi, thank you for this great talk. I was wondering, have you tried to develop models, because from what I understood, the models you developed are essentially statistical. Have you tried to develop differential equations model to understand the dynamics of these signaling pathways and to try to parameterize these models thanks to the data that you're collecting?
Yeah, so what we do is so what I present here is basically is if you want is the equivalent to a linear ODE model,
right?
Solved in steady state with. But in the past, we've actually been working a lot on more kind of really more mechanistic models in terms of ODE and time series. The problem is you have to generate a lot of. So to parameterize these models, you need to generate much more time series data, obviously, and much more data that is actually at the end doesn't really give you a lot of mechanistic insights. Yeah? So the amount of work that you have to put in experiments to get the same amount of mechanistic insights for like larger time-dependent mechanistic models for these questions, I think, is just too much.
Why do you say it doesn't give you more mechanistic insight?
It gives you have to if you have the same number of data points and you would invest it more in more perturbations instead of more time points, I think you're better off with more perturbations. That's why we are now putting focus really on that. I mean, in the past we did a lot of time series and ODE models.
I was thinking that you could make perturbations in different ways, small molecule drugs targeting a protein, RNA interference methods, gene editing. Do you see there being any potential differences in the different approaches to perturbation? For example, can you easily generate some kind of dose response if you're doing gene editing or not? Yeah. Is it all or none?
There are actually methods now for doing dose responses for gene editing. There's CRISPR tuner, which is an amazing tool. You can really titrate down expression of individual genes. I think this will be very powerful because you can also do it very high throughput. The problem with small molecules is that the throughput, you are basically limited by throughput. So I think, so my current take home in the moment is that it is actually better to do more perturbations than. And one thing that you can do is really combine drugs with genome editing. I think that's also has a high potential to basically screen the entire kinome or something and then add drugs to that and compare both the actual drug, but then also the actual drug with a different federation at the same time. So I think the combination will be a bummer.
And great talk, Niels. I may have missed that, but you have, of course, very different forms of cancer originating from different cells, which have different endowments in these pathways. Yeah. Do you use that in one model cell, or do you do that individually for each cancer cell and its origin cell?
Yeah, our group is mainly focusing on colon cancer. So we do basically colon cancer is also a class of diseases, and you see that, for example, the response to having an additional oncogene is very different in cells coming from, for example, the serrated colon cancer than, for example, the classical colon cancer. So you see that it is actually important what's the class of the classical histopathology is very important. In addition, what we see is that the additional mutation that you have in different tumors also have a large effect. So it's like there's two things, right? So really the histology, I think, is still important, but in addition, you have the additional mutation landscape that also modulates that. So it's like it's not that easy. And for what we see is, for example, that if you have a melanoma, there the monotherapies have been very successful. And so there, and then you can really pinpoint this to network wiring, where these feedbacks that I showed are not existing because they don't express these receptors. And so it's an important thing to look at which organ, the origin, and what's the genomic makeup.
Right. So we have to move to the next speaker. Let's thank Niels again.
Okay. You already realized that it's getting quite complex with the data we are acquiring. And this is a subject Sophia Forslund will talk about. Applying different omics, what we call omics technologies, multi-layers, and an approach to understand the development of multimorbidity or these inter-organ disease trajectories. And I'm happy that Sophia accepted our invitation to do so.
I am super excited to be here and to see what else I can absorb from this very vibrant environment and possible conversations. My voice is weird, so if you can't hear me, let me know and I'll try to speak up a little bit better. I'm also extremely sleep deprived because I finished these slides literally 5 a.m. this morning, so let's see how confused this gets. I'm very excited about the topic. I'm very excited about what we will be able to do together within the Interact Consortium. So just very briefly, indeed, I am affiliated with Charity and with the Max Planck Center in Berlin within this sort of overarching experimental clinical research center structure. I am a computational biologist or bioinformatician by training, and I've been trying over my career to move closer and closer to what I would call systems medicine or computationally informed, solving a medical problem rather than playing with toy examples. And we're getting closer to that because we're now able to do many more interesting measurements using multi-omics and other kind of large-scale broad phenotyping more and more cost effectively, so that eventually we will have this on the patient level scale where we can do fun data things with it. So generally speaking, my main interest is building models to say useful things about medicine from patient data. We focused a lot so far on the gut microbiome and other microbiomes as risk modulators, protective and risk factors in this. And in the course of that, we've also done a bunch of things on microbial genome scale evolution, ecosystem scale evolution, characterization in order to solve that first problem. And more relevant perhaps to the talk today, developing tools to do statistics on especially cohort level data to get closer to useful contributions without going to many of the wrong conclusions. In the concept of multimorbidity, Wolfgang already presented most of the really relevant parts. But to briefly summarize, there's so many different external environmental factors. There are internal environmental factors, which is what I like to think about the gut microbiome, that integrate, perhaps contribute to individual variability in response to these factors. There are steps and stages which we can measure in high throughput, and by throughput I mean things like thousands of genes, thousands of proteins, thousands of metabolites from an individual at once, of different kinds, and these different separate layers is what I mean by multi-omics. It means you got that from blood samples, from urine samples, from stool, maybe you got maybe genomics, maybe transcriptomics from the same individual, but you can align signals in these spaces drive eventually in the pathologies that we're interested in this multimorbid context, where various sort of components slowly start to shift outside of the allowed homeostatic range. This includes things like the components of the metabolic syndrome, your blood pressure starts to go up, for example. And then these start interacting because the body needs to compensate. As the body compensates, it starts damaging itself. This is when healthcare comes in. You start getting medications for this. There might be further follow-ups, might be side effects. And then this process escalates. So you get a vicious cycle where more and more components start breaking down. We start having to add more and more medications. And in the end, we have nothing left to do when we reach the end of the road. So because of this, we want early intervention. We want to detect who is at risk, is more or less at risk, very variable between individuals, understand causes of that, understand potentially diagnostic and prognostic biomarkers, and understand potentially which treatment can be done for each person. And here I really love the perturbation angle, because honestly, I see this as a system that can be perturbed. And if we can find for the whole body system, bacteria and human cells, what perturbation would allow it to shift towards homeostasis, then we could predict what to do to keep a person healthy, and we could even move away from the concept of singular diseases. But for now, that is what we do, and we have all these various systems that can break down separately or together, but definitely will interact. So we want these kind of biomarkers that we can use, and we try to mine it from this kind of data. The problem is that there are many different things that affect what we measure. Perhaps most crucially, there are bystander risk factors that affect different things independently. They will appear to be causally linked, but aren't. They will be confounding each other. And there are downstream effects of the disease. So something that follows because you've been diagnosed and treated is not something we can target to make you better. So if we believe that, we will do the wrong thing and we will apply the wrong treatment. Though in some cases, these are also connected. A medication might be reverting something which is part of the pathomechanism. Then we may learn from the medication where we could additionally target interventions. So there's a major need in any kind of omics level, large-scale search for biomarkers to consider the wider scope of all the different factors that in the complex environment of a human cohort, which is by far more difficult than, say, mice in a cage, that can affect these various systems and the consequences they have at the overarching level. So most of my examples today come from the Novartis cohort, an EU-funded project that we were doing, still working on this, honestly, but a lot of us come back already, trying to understand cardiovascular metabolic disease progression. Now that takes 20 or 30 years to go from early stages to late stages. So it's difficult, aside from in some very specific context, to follow someone over time. So we tried to do this sort of proxy longitudinal study-ish by following people at different stages in this sort of escalating multimorbidity. It didn't explicitly target non-cardiovascular metabolic morbidities, but as you will see, they are also in there simply because that is what the demographics book and were telling us about is yielding. For this cohort, we have various deep phenotyping from stool and urine and blood. We have very deep tracking of clinical phenotyping as well as medication records. And we've done a bunch of stuff on this already, but one thing that really came up is how heavily medicated heart patients are. Like you have individuals who are on more than a handful of different medications, and as we were identifying in other work, this is something that also clearly visible in anything you read from blood sample or stool sample. So we need to be really very careful how we account for the fact that the people we study, again, those are the people we want to understand because they're the ones we want to treat. We can't get away from this. They're very medicated. We need to understand how this affects the readouts and conclusions we get. A large fraction of the variation, as I said, in any kind of omics-based measurement from a patient is traceable to a medication, which we can hopefully in some cases turn to an advantage as a discovery tool, but it needs to be engaged with. So we've tried a few different ways of doing it, and this is old work that isn't really, I wouldn't call this AI, but it serves a purpose for something I want to tell you later. So super briefly say what we build stuff like large-scale, heavily parallelizable, generalized linear modeling framework to essentially do comparative testing of all potential hypotheses in a data set of what is a direct versus indirect effect, what's confounded versus a biomarker effect, which we think we've been clever about. So it's testing all the sort of possible associations in a naive way, and then it's doing comparative modeling, and then based on the sort of pattern of all the different possible signals, sometimes we can be certain that what we're looking at least isn't us misunderstanding something else that was in our data set. In other cases, we can be certain that it is, for example, some bacterial changes in that really are the effect of medications rather than of an EVC signature. And in many cases, we just aren't sure. But it gets us a little bit closer, and then you can take that into the experimental systems, into the single cell cultures, into the animals, and see what you can do with that. We are currently simulating this. We're building large artificial data sets, and I shudder to think of the carbon footprint, honestly, of some of these simulations because it took a lot of time to do it, but it's there. So we're pretty sure that these tools do work under semi-realistic conditions for some purposes. Still, there are definitely limitations to them. Another thing from metacarditis cohort that I want to underscore in terms of these complex patterns that can trick us when we try to learn from patient cohorts is comorbidity, premorbidity, which then extends to multimorbidity. So what we had here was we compared healthy people ultimately to heart disease patients, but then you have the middle ground of people who haven't had a heart attack, who doesn't have sort of symptomatic atherosclerosis, but who are likely at very high risk, overweight, they might have diabetes, they might have kidney function loss, etc. Now, we can find a lot of features that distinguish them from the healthy controls, yes, even when adjusting for medications, as I outlined. But by far the main, sorry, this was me confusing what I was saying, sleep preparation. My point being that there are differences between the colors, mess me up a bit, comparing the heart patients from the premorbid patients, the patients at high risk but not yet cardiovascular disease diagnosed, and the cardiovascular disease diagnosed patients. We do see signatures between them, even with drugs, mainly in the metabolomics space. But by far the most signals we see in any comparison, we see both between healthy and heart patients and between healthy and non-cardiovascular metabolic disease patients. So the premorbidity or the comorbidity is the stronger determinant of variation in the spaces we measure. So if we just look at sick versus healthy, we will misinterpret which sickness it is that causes this signal. So we couldn't develop necessarily direct cardiovascular drugs from that kind of cohort data unless we also account for the premorbidity and the shared comorbidity. Maybe we can do things over time. As I said, 20 years is a long time to follow people getting from like beginning atherosclerosis to heart attack. But we can look at things over time. Possibly, we did interventions, for example, putting people on a fasting diet trial, measuring immune cells and microbiomes sequencing it, quantifying it through cytometry, doing modeling, finding drivers of what changes when someone's blood pressure gets better because they fasted for a week and then went on to a Mediterranean diet instead of what they've been eating before. We see, for example, there are these large-scale shifts in the gut microbiome, shifts upon fasting, then reverts, but definitely in a distinct and specific direction, and correlated shifts in immune cell populations, so the overall homeostatic state of the body, which I think is really exciting because it points to the interconnectedness of the microbiome with the immune system, with metabolism, all these things interacting. We can adjust and have developed tools for adjusting this also for confounder shifts. So we do see that there are definitely improvements from a particular intervention like this. But what's really relevant to note is unknown to us, except when we looked at the records after in the questionnaires, a lot of these people had had the medication changed by the GPs during the fasting trials. So it's not enough to control for this just in cross-sectional studies. You also need to account for it in longitudinal studies. We're never getting away from a confounder problem. So we need instead to engage with it and ideally leverage it as a perturbation, actually, which is perhaps the most interesting possibility, using it as a natural experiment in getting more of the system dynamics out. So we could see, and here we also used a little bit closer to AI, machine learners to predict from the immune system who would respond well in terms of getting better from the fasting. We can see that from the immune system and also from a depletion of certain bacteria in the gut before the intervention, that's then reverted by that intervention in the ones that respond the most. We can do things like treatment response prediction from fasting in this case, but looking at how fasting microbiome changes reproduce across daily studies, this is me pulling out what people have done before. Some things show up often, more or less robustly, but I would love for this to be just straight lines across where everything is replicated in every study. No one does that in biology, obviously. This is not microbiome being worse than anyone else, but it's a problem of systems biology and it's a problem of quantitative high throughput biology. Why is this? Why should an underlying reality be fuzzy as this? It's like we're looking at somehow different things in different studies, and the question is, can that somehow be the same thing, just seen from different angles? Why are we not seeing better reproducibility between the studies? Possibly because we're looking at different things from different directions, from different modalities. But that is something we need to address. Essentially, reproducibility is the elephant in the room in the wider systems biology scope. And there are a number of reasons why I think this is so challenging in the way we've done it in the sort of one variable at a time analysis. Open too much, stay by a little bit. Yes, good. One is that, as has also been indicated already from genomics, these are multifactorial conditions with many different contributing factors to building up the whole. I mean, the body is a stochastic ensemble of processes, each of which can, in principle, work or not work in different ways. And also individuals are extremely variable. These sort of nice little star graphs I showed you, they're all centered to individual to filter out. If we didn't do that, it'd be scattered all over the place because as people, we are all by far more different from each other than we are from ourselves as sick versus healthy. So unless we find the right angle to see this from, we'll miss it. Another aspect is, in some sense, it's getting harder the better we are at making measurements, because now we can get all the genes quantified in any individual and all the transcript at the same time. So we may be able to have it from multiple cell types at once. The more we get, the more we run into a problem that we have many more variables than we have samples, because we're still limited, mainly cost-wise, by the patient samples we have. And this is another problem that needs to be addressed, I think, as a part of sort of wider scope of healthcare data sharing, making it safe for people to share that data, possibly bottom-up strategies, harmonization, integration of sort of point of care, etc. I could talk a lot about that, but I won't, because that's a different topic. But this we could also discuss in other scopes. Maybe we can found a an app or something to allow for data sharing, whatever. It is a problem with base statistical power that we are having a hard time getting around. And then that's what I said, it is about proportional factors. It's not just this thing makes you sick if it's high on this level, health if it's lower on this level, but it's whether or not these factors are balanced enough by these factors, and that also is heterogeneous. So the reality of how health arises from the multidimensional continua of the body, of this stochastic ensemble, while real, I mean, we are sick or healthy, is challenging to test, and we cannot do brute force exhaustive test variable by variable, combination by combination, triplet by triplet, quadruplet by quadruplet. And we would never be able to test this statistically at any point. We need some way of more clever searches through that data space and find meaningful hypotheses to test with the data we've got. And also on a conceptual level, I feel it's a little bit challenging that we're looking at, say, a protein being different in sick or healthy. We're not actually interested in the protein other than indirectly. We're interested in the sickness or health. So it's more straightforward in a way to think of this as, can we understand health rather than variability of the protein? And from that direction, the effect sizes also are on some level higher. So my hope now is we can go towards using more properly termed AI, or actually I prefer like impersonal intelligence, because we are also kind of artificial. We were made by other people in most cases, and well, anyways, aberrant as well. We tried a bit way back when, don't look up this paper because I had a different name at that point, which I'm not using any longer, but way back when already we were looking at type 2 diabetes versus metformin, type 2 diabetes drug, multi-omics analysis in gut microbiome data, seeing that the drug effect is larger than the disease effect. And we were testing this by building SVM classifiers, showing essentially that it's super easy to predict who gets metformin. And if you try to predict who has type 2 diabetes, what you're actually seeing is just a metformin signature in that data. I can do a little bit better now with newer models. But I think this is relevant in that you can compare different classifiers and come to different conclusions. Also in that further analysis of this data set, we can see, as I said, a lot of differences using SVM classifiers in this case. We can predict the difference between the hard patients and the healthy controls. Sure, works out well. If we try to do it, as I said, with the pre-morbid individuals versus the hard patients, prediction accuracy goes down. I told you this already, but my point is we can also see it in the machine learning space. Like if we look at not variable by variable, but what happens when we train a classifier and look at the performance of that classifier over multiple iterations of the problem, we can conclude some of the same things in a different modality. Other things done, honestly, also on the same data set, and there'll be more papers from this too, because we still keep on mining it 10 years after. It was a really good idea to establish it. Collaborators of mine in ICL were looking into kidney function, and we're able to see that we can predict essentially variation of a proxy for cardiovascular risk, so particular blood metabolite, and trace down using explainable AI which drivers in the data explain that or not, and use that as a discovery tool. What that highlights is that for this metabolite, trimethylamine oxide, main drivers that weren't perhaps all that recognized in this regard include kidney function and age, of course. But the point here is that we can see essentially how a microbially produced metabolite is impacted by reducing kidney function to then trace and increase cardiovascular disease modalities. So here is one example, actually, of multimorbidity rising in a cohort visible through dissecting machine learning classifiers on that data set. And also this reveals some other interesting insights. So we have an indication from here, for example, GLP-1 receptor agonists. You are familiar, I'm sure, with Ozempic, for example, though this is an earlier generation, seem to actually counter this process and therefore might be cardioprotective in this cohort, which would be one example of a potential repurposing of a drug, protecting against multimorbidity. And this is exactly the kind of thing I would love to find more of, because they could be moved far quicker into production. Well, not those ones, because they're not produced enough yet. So, but once that bottleneck in the supply chain is over, there'll be many more things we can do. And if we can make this kind of discovery, we can motivate that and we can bring that forward. Still, this is all sort of single specific cases. Another thing I'll mention briefly, this is also relating to what Håkan said, and this is the idea of like, you build a predictor and then you get stuff wrong a lot of the time. You try to see how old is this person? Well, this person is 60 years old, no, actually just 50 years old. Our collaborators in Israel did this in our companion paper to that one for things like BMI, for example, from the metabolite levels. But the interesting part is that the discrepancy here, the failure of our predictors, what, if you will, their silhouette, the shadow of the metabolome or microbiome, etc., cast between prediction and reality is itself informative. It tells us the missing variation, the latent variation, and that links here to diabetes risk, it links to atherosclerosis. So we can do, and there's others doing similar things, and we'll move on doing this kidney factor as well. We can essentially look at the discrepancy between predictions and reality as part of a discovery tool and potentially a personalization tool. Like some of these translate to things you can run on a person, especially with things like sharp analysis. What I really want to do now, and if I'd had like a few more hours, I would have done this to like a few more slides still, where I've dissected this in far more detail and told you how I replicate some of previous findings. In the end, not even ADHD medication and Monster energy drinks and whatever else weird stuff I put in my body helped. But essentially, the mixed effect modeling framework is driving me forward. We are now thinking that we can extend this into a machine learning paradigm. So we can do the same thing of systematic comparison of all the possible models with all the possible properties at scale, including diagnosis, including combinations of diagnosis. So therefore, multimorbidity is well represented enough in a certain category of data sets, drugs, various properties. We can see how they work, we can dissect them. There'll be a prototype out of this very soon, because probably some of you others can run with the idea, and I wouldn't want that unless you collaborate with me. So generally, if you want to collaborate, reach out. The one figure I did have time to generate before I really needed to sleep was that just looking here at the cross-prediction behaviors between different diseases visible in this data set, it's not all of them, we've got a bunch of others, but sort of the main sample ones, where you see on the diagonal that yes, a predictor is generally good at predicting the disease it was trained on, but their cross-prediction or the misprediction accuracy is also relevant, and that reflects several different things. And we can then dissect the contribution that comes from the other diagnosis or the medication, which are possibly things we wouldn't have for a new patient, but also the urine metabolites, the blood metabolites, the immune measurements. There's all sorts of exciting things we can do. And then also we are developing currently ways of robustly assessing our confidence in a given setting under careful large-scale permutation testing and other methods for what the drivers are and how those drivers may differ between different contexts and how the combinatorial effect and synergy between contexts are, which is what we're hoping to do in Interact, for example, is to extract, for example, what are the synergistic effects happening in the driver space for predictors of multimorbidity versus singular morbidity as a discovery tool. So that brings me to sort of end of it. What I'm seeing here is a new paradigm where instead of studying each separate variable at a time as associated with disease or not, in a way, a measurement-centered paradigm. Instead, we should be going to a diagnosis-centered paradigm using predictors, classifiers, and this is agnostic to what they are. They can be neural nets, they can be larger things, whatever you can actually fit to the data space. You could, in principle, do something like this with Early attempts were all like, "Ooh, we've got a machine learner. We can tell who is sick and not from a blood sample." No, you can't, because there's all these confounders. But that doesn't mean it's not useful. And also maybe eventually we can, but we should be very careful on those particular claims. But even when it doesn't work, what we can do is we can clearly use comparative machine learning, comparative AI, and explainable AI to learn not only from successes of the models, but also from their failures. They don't actually need to fulfill the criteria of being immediately translatable to making treatment decisions in order to help us understand the multimorbidity and its causes better. So as a discovery engine, we are far more capable than we are for immediate translation. And that may then later lead us to more refined models that can be used in the clinic. We still have a problem with statistical power, even if we're not doing p-value testings, but testing generalizability of models. The corresponding factor is this sort of cursor dimensionality. So we'll still need more samples, better harmonization, better controls. We are never getting away from the problem of needing domain knowledge of understanding what data we put into the model. We cannot trust it just to identify these latent classes on its own. Don't ever think about it. Maybe there are ways, but well, there's no obvious way of doing this in an entirely automated way, and you will draw weird conclusions if you do, because like yourself, your AI is a pattern matching tool. You will see faces in the clouds. You will see conspiracies in people not talking to you. The AI will see things there, but actually just reflects the very biased, in so many ways, data that you put in. And that includes sort of the obvious things, which also are aspects like the health equity aspects that I know Tura will be talking about, in who gets into the cohorts and how their variability is represented. But in addition to that, there's also things like who survived to be in the cohort, or we filtering for the people who had a certain subtype of disease for a certain medication response. Sometimes you see paradoxical output. We can't entirely automate this out. We still need to interpret the models. Very briefly then to finish up, thank you for your patience. I know I'm a little bit over time. Lots of amazing people work with me on this. I can't mention every Metacartis collaborators, there's hundreds, but we keep on doing it. Also the collaboration with the Israeli lab, and of course the fasting study, and my other Berlin collaborators, and the Interact team. I was gonna put the Interact logo here, but I didn't actually find it somewhere, and I didn't have the energy to add. But if I give something like this again at a later point in the process, I will. So that's where I will stop. We'll see. We might have questions now. I don't know what time. If you don't, talk to me later. I'll be here also tomorrow. If you want to work with me in some way, collaborate on some projects, do some kind of joint thing, whatever, or if you want to look into sort of ways of translating ideas in any kind of space, I'm open to different options here. Hit me up now or later, and we talk and we'll see what we'll find. Awesome. Thank you.
Thank you
That
very
was
much.
very cool talk. Some urgent burning questions, because I would have one. Because I would like to stress one important thing, which also was very important in the last years when everyone was working on COVID-19 samples, is to find the best control population.
Yeah.
It is not the healthy patients, because they will not tell you, and an immune response is important to any virus. I always keep telling my people now. So it is not that you have per se an immune response. It's why and how it is altered that it ends that the patient ends up in the ICU. And that's what I really liked about your morbid patients. But it's the important thing and the very difficult thing is how to identify the best control population. And how do you identify the at-risk population, which you should include into your analysis, and not just healthy versus multimorbid. This is what I really like.
It's extremely frustrating, because I think one of the things happening in addition in this cohort is that many of the diabetics without heart disease, they're actually even more obese. They're even more liver pathological, but they have some kind of protective factor that makes them not develop heart disease. So we're not even looking just at a pre-morbidity.
Yeah.
In many cases, those are women and the heart patients are men, so that's part of it. But there's all these different factors going in, and unless we're clever in interpreting, combined with clever in development tools, we won't get it.
Okay, but Sophia will be around, and
Yes.
please stay with us. We will now have a lunch break for 1.5 hours, but you can also talk to her over there. And please come back, because then we will still have 2 exciting talks addressing, as I said, the sex aspect, which I think is absolutely important. We neglect that completely. And then also talk given by Arun about one of the multimorbidity experts. So please come back after lunch or talk to them over lunch. Okay, then let's start again for the last but not least talks of our workshop. And I'm really happy to have Arun Hingurani. Is that? Well, it's actually correct.
Yeah.
On board of our cluster initiative, I have to say, and also that he accepted the invitation, because they are at the forefront using real world data, health record data there in the UK to understand multimorbidity, and this is exactly what he's going to talk about today.
Thank you very much, Birgit, and the Interact team for inviting me to speak today. So just by way of background, I'm a clinician by training with a specialty in internal medicine. So if you know anything about internal medicine in hospitals, that's the team that receives patients that none of the specialists want to take, or if they've got more complex problems. So it's always kind of been natural for me to think about multimorbidity. I'm thinking about the patient as the unit of interest rather than a given organ system. My research is largely in genetic epidemiology and in therapeutics, and hopefully I'll give you some flavor of how that all fits. I'm going to be speaking about research that has been undertaken as part of something called the Multimorbidity Mechanisms and Therapeutics Research Collaborative, which is funded by the Medical Research Council, ESRC, and the National Institute for Health Research. And I'll say something about the funding that's been injected into multimorbidity research in the UK as well, and then show some results. So I was asked to speak about sort of difficulties or challenges in multimorbidity, and I've thought about a few. The first is definition. We had a bit of a discussion about this earlier today, so I'll come on to that. The second thing is sort of quantifying the burden of multimorbidity, particularly by important strata, age, sex, ethnicity, economic factors. Patterns of multimorbidity, and we heard a little bit about this morning, particularly in random from non-random co-occurrence of disease, and understanding mechanisms, biological basis, risk factors, social and economic drivers. Polypharmacy, the role of medicines in multimorbidity, both beneficial and potentially adverse. And then how we plan our research organization, service configuration, clinical care delivery, and so on, guidelines, all our aspects of this. I won't cover those. Okay, so the classic definition of multimorbidity, as we've heard already, is the presence of 2 or more chronic diseases in one individual. And just for reasons of clarity, I'll define comorbidity as the presence of other diseases once you've defined an index disease of interest. So just to distinguish between those two. And that definition comes from the Academy of Medical Sciences in the UK. So the growing interest in multimorbidity, we already heard. Several reasons for that multimorbidity is growing. We saw some nice data from Wolfgang this morning about how multimorbidity prevalence has been increasing. It's related, I think, to the aging demography, increased survivorship from diseases that were previously fatal, but also, I think, the universal exposure to risk factors, smoking, alcohol consumption, that kind of thing, that clearly has an impact. And there's been a concern as a consequence of this that there's a kind of over-specialization in organs and systems, at least from a clinical perspective, and that we need to reset the dial and focus on individuals as opposed to individual systems. So in the UK, a number of steps have been undertaken to motivate interest in multimorbidity from a healthcare burden perspective, but also from a research and mechanistic understanding perspective. And there are a number of papers written in the last 4 to 5 years highlighting the challenge of multimorbidity, and coupled to then some research funding initiatives to stimulate activity in this area. And that's largely been led, not exclusively, but largely by the Chief Medical Officer in England, who's Chris Whitty, and happens to be a clinical colleague at the hospital where I work. So I'm going to focus on certain questions relating to multimorbidity and then see how far, at least we've tried to go with some of the research we've been doing. So here's a question that I ask medical students a lot because it struck me I didn't know the answer to this, and that is how many diseases are. I might think about that for a while and we'll come back to it. There's a lot of talk about diseases of aging, but has anyone defined that? What is it? How do you define a disease of aging? What does that look like? What's the burden of disease over life? Of course, which diseases co-occur and which co-occur non-randomly? How should we think about multimorbidity and drug development and therapeutics together? How should we think about multimorbidity and polypharmacy? And lastly, and that's the bottom bit, is really about, relates to how we should think about multimorbidity in relation to how we teach medical students about how to take medical histories and record diagnoses and so on in the medical record. So back to the question, how many diseases are there? I'm going through several orders of magnitude here. So I don't know where you want to stick your arrow. I put it around there. First thing to say is monogenic disorders, I'm setting aside. Monogenic disorders, there's probably about 7,000 or 8,000 unique monogenic disorders, Mendelian disorders. You can look up things like OMIM, OrphanNet, and resources like that. But many of these are very rare. They occur possibly in single or only a handful of families. What I'm thinking about here more is the common diseases that have complex etiology, genetic and environmental. And I've sort of centered it as a ballpark around 10,000, based on the numbers of diseases that are listed in some common coding schemes or ontologies listed there. But clearly, exactly how many there are depends on granularity. So for example, in ICD-10, there are hundreds of codes for tuberculosis because tuberculosis can affect multiple organs and there are different aspects and manifestations. Okay, so one way of trying to tackle multimorbidity is to identify data sets where all diseases are represented at scale. So where are those data sets? Well, those data sets are really healthcare systems. And in the UK, we have the advantage, and other countries have some similar advantages and systems like this, where we have a single payer healthcare system, funded through taxation, free at the point of delivery. So there's universal healthcare coverage for the whole population. It's the National Health Service. Now, diagnoses are recorded in National Health Service records, in primary care, in general practitioners' databases, and also as hospital episodes for patients who come into hospital for a particular condition. We call those hospital episode statistics. Now, that's fine, and we can identify those. Those data are available for research, anonymized. But they're recorded in different ways. So a diagnosis, the codes used by general practitioners, at least in the past, different from the coding system used in hospitals. General practitioners have used a system called the READ code system. They actually had electronic health records before hospitals did. Many hospitals in the UK still don't properly. And then hospital records are recorded using the ICD coding system. And then there are separate codes for procedures, for example, in hospital. Now, so there's a challenge in identifying those records and then bringing them together into sets of records that represent diseases that are useful for research. So a science has emerged, which is called phenomics, which is a science of developing, validating, and sharing reproducible disease phenotypes for research from very variably coded, structured, and fragmented data. And shout out here for colleagues at UCL, Harry Hemingway and First in Access, who really led the field, certainly in the UK and internationally in some aspects. So I should say there are now phenotype libraries that you can look to, and that provides a way of being transparent and open about how things are coded, and then you can reuse them. So why is phenomics important? I'll just give you a sort of kind of cartoon example here, but I think it sort of presents a message. So in the UK, we have a large population-based cohort study called UK Biobank, 500,000 people that have been recruited from the age of 40 and are being followed up. And there's genomic, proteomic, and importantly, there's linkage to their NHS health records. And so it's possible now to do genetic analysis, genetic association studies against health record phenotypes. So I don't know if any of you follow GWAS bot on Twitter, but GWAS bot is a Twitter site that basically does GWASs of ICD codes in UK Biobank and other traits, and it releases them quite frequently. So here's a GWAS bot Manhattan plot, genome-wide association study of myocardial infarction using a single ICD-10 code from linkage in UK Biobank. And you see a few hits, and they identify 6,000 cases with that code. Okay, if you look in the same UK Biobank data and use a slightly broader definition of coronary heart disease than just myocardial infarction, here using a definition that's been used in other national biobanks like FinGen, you identify double the number of cases and more hits. And then if you use a definition from the HDR UK phenotype library, which again is broader, but I think meaningful because you identify more cases and you also identify more hits. So phenomics is important. What you define by your disease is important, and particularly so in these health data sets. Okay, so how can you study diseases all at once? So I'm going to show you some work led by Valerie Pan, who did her PhD, she's now a postdoc, using linked health record data. So in the primary care, it's possible to get a data set from the Clinical Practice Research Data Link, or CPRD, well, in this analysis, it was 4 million people. We now have a data imprint of 20 million people from the same primary care data. And it can be linked through NHS Digital with hospital episode statistics and mortality. So you can generate a kind of synthetic cohort by linkage of these electronic health record data. And then you can start to answer some of the questions that I was talking about. So based on phenotyping phenomics of 308 physical and mental health conditions from 4 million people, it's sort of possible to examine the prevalence of different diseases at different stages of life, because people at all ages are represented within the cohort. So that's been published in Lancet Digital Health some time ago now. Look at that if you have interest. And then this idea about defining what an aging-related disease is. So it's possible using these electronic health record data to generate age-specific incidence rates for each of the 308 diseases. And you see patterns that define age-related diseases. So if you look at the top, a clear age-related disease is age-related macular degeneration, very common degenerative disease of the eye that's a major cause of blindness. And you can see that the incidence increases with age, but actually the age at which you start to see an increase in incidence is fairly late. Contrast that with hypertension as a diagnosis, where it's also clearly age-related, but you start to see it pick up from much earlier. And then you have two other conditions there that are clearly not, well, age-related, but maybe in the opposite direction: appendicitis, bottom left, and depression, where you can see this kind of peak in young adulthood and then again in later life. Okay, so then coming to this question of co-occurrence. So we felt it's important to distinguish diseases that co-occur more commonly than you'd expect from their individual frequencies, because they might share some common mechanisms, from those diseases that co-occur in line with their individual frequencies. So on the right, I've shown in this individual four diseases. On the right, 2 diseases, I've labeled them P and Q, where they're observed to occur, P and Q together, in line with the frequencies that you'd expect from those individual conditions. Whereas on the left, A and B, 2 other conditions, are observed to co-occur more commonly than you'd expect from their individual frequencies. And you can start to think about why that might be. It could be that A causes B directly. So it could be that atrial fibrillation causes cardioembolic stroke. It could be that A causes B because a medication for A causes B. So atrial fibrillation might be treated with anticoagulant drugs, and B might be subdural hemorrhage, a bleeding complication of anticoagulant drug exposure. Or it could be that A and B share a common cause. So hypertension might be a risk factor for atrial fibrillation and ischemic stroke. Identifying diseases that are non-randomly co-occurring might provide a route to focusing on the ones that you might want to try and investigate a mechanistic basis for. Now, that's led us to kind of propose an expanded definition of multimorbidity. And the idea is that because chains of causation could be separated in time, that if you're interested in mechanism, you might need to extend the definition of multimorbidity to the ever occurrence of 2 or more conditions of the same individuals, regardless of chronicity, and look over the whole life course. So there are examples of this. So we now know that Epstein-Barr virus infection in early life is a risk factor for developing multiple sclerosis. The treatment of certain childhood cancers is a risk factor for vascular disease in later life. Preeclampsia is a risk factor for vascular disease in later life. So unless we know the whole life course, we couldn't probe those issues. Okay, so in subsequent work, we started to look at co-occurrence and non-random co-occurrence for the 308 conditions that I mentioned earlier, and that's published in a more recent paper. And so here you take comorbidity relationship. So this is age-related macular disease, and then this is simply the prevalence of other conditions given an index diagnosis of age-related macular disease. And then here for age-related macular disease is the non-random co-occurrence of other diseases if you have age-related macular disease. So we've done this for the 308 diseases. It's possible to use that information to construct disease networks. And what we've done is develop some interactive tools that you can go to investigate co-occurrence, random and non-random, from these data. So this is a screenshot of our website. If you Google MMTRC, you should be able to get there, and then something about the whole program and about the tools to probe these data. Okay, so if you've identified diseases with common mechanisms, you might start to ask why. And it could be that 2 diseases are related because they share a genetic basis, or it could be because one causes the other. And so there are genetic approaches that can be taken to dissect that. So if those diseases those pairs of diseases have been studied by a genome-wide association study, there are techniques for investigating genetic correlation, and there are techniques for investigating causation using Mendelian randomization. But of course, you might have other plausible and strong hypotheses about common mechanisms that might lead to multiple diseases. For example, immunological factors, inflammatory factors, metabolic, and so on. In which case, you can take a kind of molecule or system-first approach, and again use things like Mendelian randomization to understand whether those types of exposures or individual candidate exposures are causal. I'll start to show some examples of that. So we think genomics is important here for understanding mechanisms. And the reason we think it is because if you're trying to understand the causes of disease, you can reduce it often simply to the task of knowing which proteins are involved in that particular disease. And one way of trying to ascertain that is to do a genome-wide association study, because by design, you're interrogating the involvement of every protein encoded in the genome for that disease because you use variants in the gene encoding those proteins and test for their association with disease. And the advantage of that approach is that you're using the correct organism, the human, you test all targets concurrently, and genome-wide association studies by design have a low false discovery rate and typically replicate before reporting, before publishing. But another aspect of it is that genetic variants are allocated at random at conception, just like treatment allocation in a clinical trial. So genetic studies are like natural randomized trials. So effectively, if you have a variant in a gene that encodes a protein that might be a drug target that affects its expression or activity, you might be able to use that information to infer what would happen if you designed a drug against the same protein. You could infer the downstream effects of that drug. So we've done this using many like examples. So then sort of thinking about this much more broadly and in relation to multimorbidity, I've already said that there are 10,000 diseases, ballpark. We know that there are 20,000 protein coding genes. Now that's a simplification because we know that each gene might produce multiple splice variants and then different protein products. We also know that functional proteins come together as subunits to generate larger macromolecules. So there's more diversity and complexity there, but I'm simplifying. So if we imagine there are 10,000 diseases and 20,000 protein coding genes as a first step, we have a sample space of 200 million protein disease pairs. And I've just shown an inset of the first 100 diseases and 200 proteins. Now you can imagine then the diseases are the columns and the genes and proteins are rows. So if you do a genome-wide association study, you're taking one disease and you're interrogating all the genes and proteins involved in that disease, and you'll come up with some hits. Now, you might be interested in drug development in targeting proteins that you think are druggable. So the thin horizontal blue lines represent the genes and the proteins that are given that are thought to be druggable from the druggable genome, if you like. So this protein is causal in this GWAS, and it's also one of those druggable genes. So that might be one you want to work on. And then if you do multiple GWASs across many diseases, that same protein is often found to come up as a hit in multiple conditions, so-called genetic pleiotropy. So then you start to get a readout of what modifying that target might do from a drug development perspective. Now, that's theory. We've done some sort of calculations to think about how many diseases might perturbing a single target affect. If you assume that there are 10,000 diseases and 20,000 proteins, and if you assume that there are, I can't remember in this calculation, I think we said if we assume there were 100 causal genes or proteins per disease, then you can ask the question, how many targets might be how many diseases might be affected by perturbation of a single target? So the calculations, depending on what parameters you put in, are that perhaps 50 diseases might be affected by perturbation of an individual target. And for repurposing, you might ask the question, what's the probability that 2 or more diseases share at least one causal gene? This actually uses a classic problem in probability, which is if you have a room like this, what's the probability that at least 2 people share the same birthday? So it's the birthday problem, if you know that problem, probability. Anyway, if you take 8 diseases, even if you assume they're independent, the probability of not sharing at least one causal protein is actually very low. In other words, the probability of sharing at least one is quite high. So if you share proteins across diseases, then that suggests that repurposing or indication expansion should be possible. And in fact, we know already that there are many targets that are, for which we have drugs that are used in multiple indications. I just give a few examples here, but there are really very many, and there are more and more. Big interest in these GLP-1 receptor agonists, these magic weight loss drugs that are also able to treat obesity, heart failure, kidney disease, and metabolic dysfunction-associated liver disease. And the genetic evidence suggests this too. So this is data on the interleukin-6 receptor. So in a genome-wide study of rheumatoid arthritis, variants in the interleukin-6 receptor were found to be protective. And in fact, there's a drug that targets the interleukin-6 receptor, tocilizumab, that's already used in the treatment of rheumatoid arthritis. But in other genome-wide association studies, the same variant in the interleukin-6 receptor comes up for heart disease, atrial fibrillation, abdominal aortic aneurysm, not shown here, but also inflammatory bowel disease. And so you can start to get a readout of what perturbing a target might do from human genetics, both from an efficacy perspective, but also from a safety perspective. So you can see in this analysis, the risk of asthma appears to be increased by variants that are associated with reduced receptor signaling. So this work and this type of principle has been taken forward at scale by Mike Pietzner, Claudia Langenberg at the Charité and Queen Mary, and done essentially across the genome, adding in proteomic data that's now available to try and essentially to construct these proteogenomic maps, proteins that are involved in different diseases and where the overlaps. So I'm just going to sort of come to the end and on the subject of use of medicine. So this is not drug development, it's thinking about medicines that are already used in people with multimorbidity. And this is sometimes referred to as polypharmacy, which simply means receiving multiple medicines. Now, it's often used interchangeably as something that's bad, but I think that's not necessarily the case. If you've got multiple diseases, you might need multiple treatments. So appropriate polypharmacy occurs when the patient receives all the medicines appropriate to their problems. Insufficient polypharmacy might be a situation where the patient receives an insufficient number of medicines appropriate to their condition. So some therapeutic options have been overlooked by their physician. Inappropriate polypharmacy occurs when patients receiving medicines that don't relate to their problems, may be irrelevant. And then there might be another aspect of this, which is a therapeutic clash, where a patient has two conditions that are not mechanistically related, where a drug is indicated for one of those diseases, but is contraindicated for the other. So an example here might be a patient with atrial fibrillation and cirrhosis. An anticoagulant drug might be indicated for the atrial fibrillation to reduce the risk of thromboembolic stroke, but it would increase the risk potentially of bleeding because of cirrhosis. So how do you identify those things systematically? And then the more medicine someone has, the greater the risk of an adverse drug reaction and of drug-drug interactions. So one thing that we're starting to propose is that certainly in the medical record, medicines and diagnoses should be recorded together in a kind of matrix format. So you can look across the medicines and see whether there are appropriate diseases for those medicines. And you can look down the columns, which are the diseases, and ask, are all the medicines indicated for that disease being used? And are there any discrepancies? This is something that we're starting to go through now. I haven't got any real data to show you on this, but we're looking at this both at an individual patient record level. Traditional medical teaching is that you record the medical diagnoses or past medical history and the drug history in separate places, which seems completely illogical. Okay, so to end and summarize, so multimorbidity is achieving greater attention, quite rightly. There are new methods that are able to unlock information contained in research cohort data and also in electronic health records. Important to distinguish diseases that co-occur simply because they're common from those that co-occur in excess of their expected individual frequencies, because they might share a mechanistic link. And I think there are theoretical and empirical evidence to support the idea that indication expansion and repurposing could be done much more widely. But clearly, from the range of activities I've shown here and also the research and so on that we've heard this morning and we'll hear more of later today, multidisciplinary approaches are definitely needed to tackle this issue. So thank you. And collaborators and investigators are all listed in the Multimorbidity website. So you'll find out who did the actual work.
Thanks a lot. Questions? Oh,
Yeah.
yes.
Do we actually see diseases that occur less often than by chance? Or is this difficult
One is protected
statistical?
from another,
Yeah.
potentially. I can't remember, that's possibly the case, I can't remember, yeah.
But in those cases, many of those is probably about a person we both would be dead.
Well, possibly.
It may not be a protective factor, it could also be just a
It
population.
could be a competing
filter,
risk. Yeah,
Yeah, it's really getting difficult.
It's not really a question, it's more of a comment. So I'm coming from the rare disease space, and we are actually moving forward in thinking about, because in rare diseases, patients are very few and very scattered.
Yeah.
But they do, many of them maybe share the same molecular path.
Yeah.
And so there is a thinking to also do trials, to do research in looking them, grouping them together by mechanism, by mutation in this pathway.
Yeah.
Called share molecular etiology. And
Yeah.
so this is actually really much echoes that. And
Yeah.
there are some trials in pushing forward in that direction too.
And I think that's right. And actually even comparing rare and common conditions where the phenotype, you might have an extreme phenotype that's caused by a single gene defect, but a similar phenotype is seen commonly in the population, but perhaps less extreme. And when you compare the findings of genome-wide association and linkage and sequencing studies, sometimes you see rare variants in the gene cause the extreme phenotype, and common variants influence quantitative variation in the same traits. I think there's a lot to learn from going across those facts here.
Further questions, because we have just one last talk following afterwards. If not, then I have a question. I mean, I'm not a genetics person, so I always challenge the GWAS people with can you also get, or can you miss something? You know, what is your thought about missing something or getting into the wrong direction? Because if you have an polymorphism in some of the genes, due to some other polymorphism, you might be protected from developing the disease, and that so gets you into the wrong direction with all your analysis. How do you deal with these?
So I think the failure to find a genetic association with a particular gene, and by implication protein, in a particular disease should not rule you out from working on that protein for that disorder. Partly because there could be, people have argued, adaptive mechanisms that sort of suppress the effect in early life of a deleterious variant. And obviously those mechanisms are not in train in later life when you might give a drug to target the same mechanism. So I think the strength in the genetic analyses is follow up on the positive findings, and there's plenty of those to be working
but how do you find out that there is maybe an interaction between the pathologic and the protective polymorphism?
Yeah, so it's sort of really a question about gene interaction or gene environment interaction. So identifying those is challenging, partly because the sample sizes needed are even larger than the sample sizes you need for the main effects. When people have done this for the really large GWASs for continuous traits where you've got most power, like body mass index, for the most part, the model that's emerged, and I think is kind of held across multiple diseases and traits, is that these effects at different loci are more or less independent and additive. Now, some people have argued that that might be simply because of the way in which we do genetic association analyses, but the model fits an independent additive effect for the most part.
I will finish, but because I think, you know, identifying these protective ones could also help you to identify therapeutic targets.
Right. So I think in a GWAS, you do identify variants that both increase risk or reduce risk, or increase the level of the trait or reduce it. From a drug development perspective, following up on those things, typically in drug development, you try and develop drugs that either inhibit or block rather than activate or stimulate, because those types of drug mechanisms tend to result in downregulation. But that doesn't mean you need to rule out that particular mechanism, because it may be possible, for example, to find an endogenous inhibitor or activator of that protein that you can then target in an inhibitory way. So a lot of kind of computational biology, bioinformatics, and experimentation might be needed to follow up on this.
I think if you would have a good machine learning model which really captures these effects, then explainability could kind of distinguish between or differentiate between the factors which contribute positively to the risk and the ones which maybe inhibit the risk or reduce the risk. So, but I think in the first place it's maybe tricky to train a good model which really captures this effect, because the data is scarce and the dimensionality is very high.
Yeah, and I think aspects that we've started to look at is that you bring in information from other fields and other data sets. So you might have bioinformatic data on where a gene or protein is expressed. You might have drug effects and things like CMAP, and you can kind of you could build a network or graph incorporating those effects that you can then start to interrogate using AI approaches. So I think there's much scope in different aspects of this for AI and machine.
Thank you very much.
And we will now conclude our workshop, and I hope you enjoyed it. With a talk given by Turu Stadler, who will touch upon what I mentioned already several times, gender aspects in multimorbidity or in health research in general. Thank you.
So let's see what I can add to this. There was already so much mentioned, I want to thank my team in gender and medicine, and thanks to the understanding heterogeneity team who have also contributed to this, and I want to acknowledge our funding. Now, basics first, right? That's why this is called Zero. Why do we need this? So why do we need gender sensitive prevention? So basics, like you know all that, I guess, but so biological sex, sex chromosomes and sex hormones, of course, are related to disease pathophysiology, manifestation, and response to treatment. But we have pretty good data by now that more gendered constructs that are more socioculturally shaped, like lifestyle, nutritional habits, exercise, perceived stress, smoking, that are usually very malleable as well, or malleable once you're not addicted to smoking, for example, as prevented. So they're probably more related to disease perception, help-seeking behavior. Men are terrible at seeking help, for example, they also cost much less in healthcare systems, yeah. But they usually, when they come, then their disease is often much more progressed, and that's probably not biological. Yeah, so there is more societal and gendered reasons for that. And perceived stress levels and then use of healthcare is gendered. Decision making is gendered, and the therapeutic response. And so we know, and it's always funny, so I'm always being asked, so Turu, which one is it? Is it biology, or is it sociocultural factors, the environment? And I'm like, have you not had your genetics? Yeah, so it's always both, right? So this is completely the wrong question. It's not either or, but an interplay of both, right? So we know that we have epigenetic effects of environmental factors, and kind of like any genetic expression is always shaped as well by an environment. And so when we talk about sex and gender, we often look out to our patients, right? But we also need to look in at us as healthcare providers. Yeah, so we also have sex and gender, and that shapes how we interact with our patients. Here we have now, so the burden of disease is gendered. And so here, this just came out, which is very handy for me, so I don't have to look that far. So maybe I just show it for high income countries, but we see that it's a global phenomenon. I'm just gonna, so I can point here. So we see that, for example, there's especially high burden of disease, where we have much more disability-adjusted life years per 100,000 persons in the population in high-income countries, for lower back pain, for depressive disorders, for headache, anxiety, other musculoskeletal, and of course dementia and Alzheimer's. And so we see that we do see that there are so gender medicine is really about women and men and non-binary people. Yeah, so we have although for non-binary people we have very little data. Yeah, so that's why I'm going to be very binary in my data presentation here. But so we see that men are very often strongly affected by this burden of diseases, and it is gendered as well. So, yeah. So, for example, COVID was probably the poster child now for a disease that the men had worse trajectories, worse outcomes, a higher mortality. It was also very good for gender medicine because it raised attention. Yeah, so that sex and gender do play a role. Here we have the burden of disease again. But maybe I already made my point, I think, so I can continue. But maybe actually what is important is to kind of see that there are areas where we have very strong, for example, Alzheimer's disease it's really interesting that it's very strongly skewed so that it's much more important for women as a higher in the hierarchy. Here we have now, so we are all about multimorbidity, right? So here we have a very interesting analysis that Schäfer and colleagues did with claims data of patients over age 65. And so they clustered the diseases. And so we have they found a cardiovascular and metabolic disorder cluster, and then anxiety, depression, somatoform disorders, and pain, and neuropsychiatric disorders, and then looked at the overlap of these. These are older, so 65 plus, but it's kind of interesting. So we see for the female population, yeah, so about half of these 65 plus patients do not belong to any cluster, and for men that's the same, as we have women here and men here, and so we do see that the overlaps, or simply visually we see that there is differences in this how these are expressed in this mostly Austrian population, I think. So, but in the end, we don't fully understand this very well yet, right? So yeah. But what the good news is that a lot of this mortality from non-communicable diseases, and this is mostly non-communicable diseases, is avertable. Yeah? So and here we see for both female and male participants, like in the darker gray, across the age range, there's large proportions of the percentage of deaths would be avertable. We could do something about it. What would that be? So what can we do? So it's always the same thing. That is also interesting. It doesn't matter if you look at the dementia or you look at cardiovascular disease. It's pretty much always the same things that we should be doing. We should be having a better diet, manage blood pressure well, manage our weights, look at cholesterol, but then things that are also very malleable: physical activity, smoking, and alcohol. And so we see that across different global regions, we have pretty much the same risk factors. They vary a little bit in the hierarchy, but what is important is usually pretty much comparable. So we know what we should do, and just turning this around and in a positive way, maybe. Here's this very nice study by Guillen from the Veterans Affairs. So 8 behaviors can add up to 20 plus life years. Yeah. So eat well, avoid cigarettes. Cigarettes alone, so if you have kids, yeah, keeping them smoke free, up to 10 more years of life. So get a good night's sleep. We just discussed this. We're very bad at this. So be physically active, manage stress, because stress cascades into other conditions. Then avoid binge drinking, be free from opioid addiction. Unfortunately, we had to add this to the list. Yeah. So, but this is very serious because in the healthcare system, we need to manage the pain medications much better. Yeah. So and have positive social relationships, because they are as important as other health behaviors. Exactly. We manage that in the Interact consortium. So here, very interesting description. We've known this for a very long time, but it's very nice to see it playing out again. So ideally, the earlier you start with having all 8 habits, yeah, so that the more healthy life years you're gaining. But it even pays to start with at 70, yeah. But so the more of these low risk healthy lifestyle habits you have for male participants here and for female participants here, the more prolonged life expectancy you gain. So, but this is not easy, and we know we are usually very far from this, as we have just demonstrated that we have had long nights, arriving late, and so on. So we did an intervention. I did that with a health insurance association. That was my PhD. So we followed 266 women between 30 and 50 for 2 years. Yeah, and so half of them got just information and a little bit of tracking of health habits. And then so here we have the fruit and vegetable servings per day. And so they all started at as I'd say, like maybe 22, 23. And if you think about your 5 per day, that should be 35 up here, right? So we all are far from optimal because it's really hard to eat that much fruit and veg. So women know if they're motivated, they know how to increase fruit and veg intake. Yeah? So all of them, the ones who were just informed they should do this, also started doing this. And then what we see is that the group that just got the information, these were motivated participants, they go over the 2 years, they go back to baseline. And the ones who got this intervention, that was really like, was the olden days before we had any AI tools, but really personalizing with prompts and diaries and so on. So to keep the goal salient, and it basically make plans and adapt those, develop personalized strategies to eat your fruit and veg and be physically active. And we saw that was pretty successful. Yeah? So they were maintaining that in the self-regulation group over 2 years. Of course, 2 years is short if we think we need to do this over a lifetime, but well, we'll see. We'll do studies over at least 7 years. Yes. So now, of course, fast forward to more interesting digital interventions. So we did a personalized digital intervention for people with very high alcohol consumption. And the intervention were personalized text messages, and the control was weekly assessments. And here we see the control. So here was the level at baseline, 24 drinks per week. That's a lot of drinking, yeah? So way too high. And then, so the control group shows a little bit of improvement here in our pilot study, 152 participants, and we see that this very cheap intervention brings down the drinking in this high risk group. Yeah, so we did try out different things. So tailored adaptively was best, and that's what we then used in our fully automated randomized controlled trial with 723 participants, 2/3 female. And this was interesting. So the whole thing was fully automated. Yeah, so recruitment was fully automated via social media. And then the whole, we followed them over 6 months, was all completely automated. And we had increased interest in the study because it was the pandemic. A lot of people were drinking a lot more than they should be. And with this intervention, we seem to be reaching people who really drink secretly. A lot of their social environments don't seem to know how much they're drinking. And so here's what we are finding, and that was really interesting to me. So here's the level at baseline pooled across baseline levels. And we see that even the weekly drink tracking also had a very nice effect. Yeah? So that people are really keeping that goal of reducing their drinking salient. But of course, our tailored adaptively does a little bit better than the others. So that's very nice for us. And then at the moment, we are running a study in Berlin schools. We have 32 schools involved in the study at the moment, currently over 1,000 students recruited. And so we look at risk factors for male and female students. Yeah? So we do gender-sensitive prevention with them. And so what was very interesting in the pilot study is that the boys, so of course, this is highly overlapping distributions, yeah, but we have in the pilot studies, we found that boys are tempted to smoke when they want to be cool, when they want to be part of the gang, kind of. And the girls think about So being more prone to smoking when they are really stressed out and want to forget about everything. And so we took great care to have have content. Of course, we didn't split them up by any gender subgroups, but we had personalized prevention strategies that catered to all of these motivations. Yeah, so personalized planning, role playing, and then self-confidence training with an actor. How do I come across as strong and convincing, strengthening self-efficacy and refusal skills? And we have sports people, like so Olympic rowers, I think, and then influencers and this actor on board to help these kids realize that they can they can be cool and smoke free. Now here it's interesting, what we can do with digital tools is very limited. Here we have a real barrier because we can not work with them until they are about 14 years old. And so that's for privacy reasons. So they are all on WhatsApp and have all kinds of digital tools, but we cannot reach them. So we are really looking forward once they turn 14, we will work with them and accompany them digitally. So these were a few examples of personalized interventions from the olden days to now, and now what we can do in the future. But it will be absolutely necessary to close the gender and diversity data gaps that we are having at the moment. So we just had a spring school about understanding heterogeneity, and our understanding of heterogeneity is currently very limited by these data gaps. And so we have, it's not routine to incorporate thinking about heterogeneity when we develop our research questions, and really, so to try to recruit heterogeneous samples. A lot of our samples are weird, yeah, so Western educated, industrialized, rich countries in democracies. They're not globally representative, of course. We are not really designing studies to understand heterogeneity. We are not used to that. The recruitment of heterogeneous samples and stakeholder involvement is costly, and that's why we often cut corners there, but we shouldn't do that. And the ethics and the data security is grueling. Yeah? So we just did a study of the Charité Stärken, where we try to describe the working climate and also the diversity of the staff and students. And it took us 2 years to get that through data security. Yeah? So it's, through ethics. Yeah, sampling is of course an issue, and then assessing heterogeneity in psychometrics is a challenge, and then data analytic approaches. So we need to get much better. I saw many nice examples today, but I think we really need to, as a research field, get better about that. But what I want to talk today is so we can do all that and we can become better. But one basic thing is AI will never help us if we do not, what's not in the data, we cannot get out of it. And so here's what about gender and diversity. Some diversity domains are routinely assessed and measures are able to cover the spectrum, but individuals situated at the margin are not included. Yeah? So kids, for example, or older patients with multimorbidities. Yeah? So when I study a certain drug, then I usually use a homogeneous sample of patients who have only one condition, yeah? Not a multimorbid sample. And children and adolescents are usually excluded as well, as are women during pregnancy. Then some diversity domains are routinely assessed, but we lack inclusive measures such as gender. So we have usually very binary conceptualizations of sex and gender, and so we should probably do better about those. And then some diversity domains are hardly assessed at all, for example, care responsibilities. To understand why a lot of patients, for example, refuse to take up rehabilitation services that are really important after, let's say a myocardial infarction. A lot of them say, I want to take care of my family, and then we really need to work with them. So we need to close these gender and diversity data gaps. And so here are first ideas what we can do. So I spent quite a lot of time in the US at Columbia University and then in Aberdeen in the UK. And I was struck when I came back, like how big the data gaps were in Germany. So, and then we drummed up this group to figure out like what could we do to address these gender and diversity data gaps systematically. So we came up with this, so this is the group 54 experts, some from the Berlin area, but also international experts. What should we actually assess when it comes to diversity? Yeah, so of course, sex and gender, we should always do, and we do, but in this limited binary way. And then age, usually we also have. Yeah? These are the two that we usually have. But then we know that socioeconomic status has massive effect sizes, but we also very rarely routinely assess it. Then care work and also integration in the labor market. Then mental health, physical health, and disability we should probably also routinely assess. And I really like what Aaron said. Yeah, so usually when we have a focus disease, yeah, so we are often not so particularly good to also characterize the other comorbid diseases. So I think that's quite important. And then we have our problem zone, which is for historical reasons, yeah, through the Nazi dictatorship and also the Eastern European and Eastern German dictatorship, massive problems to systematically assess sexual identity, ethnic racial identity, and religion and worldview. So the issue is lots of racism can't talk about race. So we need to earn the trust of our populations to really assess these things so that we can personalize treatments for them as well. So here is now, yeah, so we've developed it first for Germany and then translated it for created country working groups. There's currently over 90 experts working with us on this diversity minimal item set. So one, I can't point, yeah. So on the left side, you see the example for gender in the US. And then you have the example for Spain, translated to English, and then also in the main local language, which would be Spanish. And then so here you see you have gender. And so we have woman, man, and then you have many other additional gender identity options to identify with. What I find really important is that there's always room in all of these items that we have a gender identity not listed here, and then people can specify, so it's a learning instrument, and that you always can say, it's none of your business, I don't want to answer this, prefer not to answer. So what do we think this could be used? Yeah, so definitely to improve the data descriptions, like, so what are our AI tools actually based on? Yeah, so they have very sharp and I would think limitations, and we should be very upfront about those. Yeah, discuss the generalizability of our research findings and the limits of the applicability of our AI results. If a result has come mostly from a male database because we are using military data, then we should absolutely need to say so. So it will have blind spots. Then, of course, we can use it to pool data systematically to detect smaller effect sizes and reach critical sample sizes for smaller marginalized groups, and then to develop personalized and more innovative interventions that tailor to the needs of diverse populations. And that was first what I thought, that's it. Yeah, so research we can do better. But then we found that actually this should also be applied for clinical practice. So to allow patients to self-identify on gender and diverse domains that matter for their healthcare, because only if we know about these things can we personalize the care for them. Then for policy development and policy impact analysis, we were to develop AI policies with balanced costs and benefits for all and analyze the impact of them on diverse population subgroups. And then, and this was a little bit controversial in the beginning. Yeah? So should we apply this also to organizational development? And I think, yes, we need to know how lived experiences and diversity domains are covered at each organizational level. If we have like a lack of skilled labor and we are hemorrhaging talented people just because they are women, just because they're people of color, then we are really losing talent, and we need to know about that. Now, but this is very important. That's very, like, so I was presenting this at a meeting in Frankfurt, and then one medical doctor said, Frau Stadler, like, my sexual orientation is none of your business. And I was like, absolutely, it's absolutely not. Yeah, so we want to do this on a organizational level and really safeguard the individual personal privacy. That there should be no forced outings and exposure. Yeah, so there we have found workarounds by doing automated assessments. So yeah, we do this now for 20 countries. And we need to earn that trust, I think. And here's this why is, so we need to kind of like make sure that it's safe for people to trust us with their with diversity information. Yeah? And so there are a lot of things that we can do to improve that. One of them is really also working carefully, using actually technology to automatically collect the data and then delete them when we don't need them anymore, and look at only aggregated levels. And also say, yeah, so there's lots we can do. what can go wrong? So there can be unintended effects. Yeah? So especially if we use biased data to train our AI. So I'm just bringing this example because it's a very interesting one. There was a race adjustment in the calculation algorithm for estimated glomerular filtration rate. And so there was this assumption that African-American patients have higher muscle mass, and it's related to higher creatinine levels. Now, this was just removed, because it was based on too small a database, and it really hurt African-American patients. It underestimated their kidney risks, despite them having a higher risk of chronic kidney failure. So we really need to rigorously test if personalization has indeed the intended effects. So I think there's a lot of promise for doing this, and only so now. Exactly. So if we want to really personalize, we will need AI because these are complex decisions. And so I hope that by getting more diverse data and really do that systematically across many different studies, we'll be able to succeed and get more AI for good. Thanks for your attention.
Thanks to questions.
Well, first of all, thank you. I just took a photo because I have to adjust my teaching slides for chronic kidney disease. Because that's still in a lot of data that you have to adjust for race.
Yeah.
And second, you pointed out the 8 behavioral steps for longevity. Do they differ between sex and gender? Is that known? I mean, is there things that women versus men versus non-binaries preferentially profit from?
So this is not fully, I think the jury is still out on that. Like, we know that smoking seems to be especially bad for women, but smoking is one of the biggest preventable risk factors for everyone. So there are differences in what we see, so that men, for example, have a higher red meat consumption, for example, higher saturated fat intake, while women eat more fruit and veg. So there are known differences that lead them to, for example, greater obesity in men. But we do also know that men have higher ambition for physical activity, so they are more easily motivated to become physically active. But they, in overall, we are all pretty bad about that. Yeah, so there are differences, and I think fully we don't have that much really good data, for example, sensor-based data for physical activity that would fully answer that, I would think. Yeah.
Super interesting as always, and lots of things to sort of discuss and think about further. I have one happy comment and one question of another area. So the first would be the interventions and the compliance and the differential compliance. I think that's a super major confounder in studies similar to what we saw with the drugs, but also with dropouts, as was discussed. And I think this is something we are nowhere near monitoring the way we should, and we could really use your help in doing so. Also, to explain some paradoxical findings where patients following like a stroke or a heart attack ends up being healthier than the people immediately before because they've gotten changes. I don't know if this is something that you've seen in data. So that's one area of curious in thoughts, and the other is with regards to one potentially major dimension perhaps of phenotype that we're not recognizing, but that could be acting indirectly through almost all the other socioeconomic variables, is chronotype. So morning versus night person, but also subclinical neurodivergence of different kinds. And I thought those wouldn't be crucial. We wouldn't be missing much with them for somatic diseases, but then I'm realizing you have all these lifestyle factors, and I just look at how much I, as a night person, am affected in different ways. So like Do you see what I mean?
Absolutely. Like, so for anyone who has a later chronotype, they are living in a toxic environment, right? So we are really, so there is discussions, for example, for school children, if you would start school later, and especially when puberty hits, like, so people start developing a shifting their chronotype, and would be, so you would even see learning improvements and definitely mental well-being improvements, more sufficient sleep times. So I think we do need to change the toxic environment around that. And I agree, like a lot of these lifestyle things are also correlated. Yeah, so that's why it's a little bit sometimes deceptive to report them one by one. So it's interesting to look at them as a conglomerate.
No further question. Probably everyone is thinking about the input they got today. So I hope you all enjoyed the workshop, and wish us luck for the application so that we can really start our research on the interorgan trajectory, so to multimorbidity. And thank you very much for your questions and for being here.